So hello everyone. It is a big pleasure to be among you, the speakers in this nice conference. Thank you for organizing it, and thank you for inviting me to participate and share my research results. So in the background, you can see the massacration of Cyanobacteria in Baltic Sea. But these blooms are, of course, occurring worldwide in freshwater environments as well. And they definitely are aesthetic problem. If the water looks like this, you don't want to go to swim in it. Cyanobacteria are known to also produce odorous metabolites, geosmin and methyl isoborneol, which are causing this muddy, earthy odor and taste in water and fish. But they are also well known to produce toxins. And of course, when this kind of massive blooms are toxic, they are very big health risk for human beings and animals. Plenty of animal poisoning cases has been reported. There has been dogs and there has been cows and there has been also wildlife poisonings as well. And we know that there are two major types of toxins, hepatotoxins and neurotoxins. So here you can see the chemical structures of cyanobacterial toxins. There are these hepatotoxins, which are cyclic peptides. As you can see, this picture is of microcystins, but then nodularin is very similar. It's only missing these two amino acids here. It's pentapeptide, while microcystins are heptapeptides. And you can see that there is special feature, this unusual amino acid ADA, which is found only in these cyanobacterial toxins. There, in this picture, you can see these neurotoxins. There is this anatoxin A, anatoxin AS. You can see that it's an organophosphate type of compound. And then saxitoxins. These saxitoxins are produced not only cyanobacteria, but also dinoflagellates in sea waters. Since these are hepatotoxins, these are neurotoxins, they block the neurotransmission in way or another. Anatoxin A being very prominent preventing neurotransmission. Same is true with anatoxin A and also saxitoxins. We had a very big cost action where almost all European countries were participating, and we had this toxic cyanobacteria as a topic, and we published this book, Handbook for Cyanobacterial Monitoring and Cyanotoxin Analysis. If you are interested to learn more how to sample, how to analyze chemically the toxins, this book might be a very good source for information. So as I said, that we know the main toxins, but new toxins are continuously being recognized. We found this kind of benthic from the benthic strains growing in the Gulf of Finland, coastal areas. This kind of compound, again, cyclic peptide with a long, very long fatty acid tail. And now we know at least 4 different molecules of these compounds. And when we were cultivating, growing it, we saw that most of the toxins are kept inside the cells. It's the same is true for also microcystins. And these new toxins turn out, we call it anapinolysins, and they induce cytolytic cell death of nucleated cells, hemolysis of red blood cells, echinophyte transformation, and are potent detergents of cholesterol-containing membranes, eukaryotic membranes. They don't affect prokaryotic membranes. So new toxins are found. Very many years ago, we also wanted to know which cyanobacteria are toxin producing. In that time, the only way we could verify this was to isolate cyanobacteria for cultures and study their toxin production. And this yielded for us at the Helsinki University large culture collection of plantic and benthic and even lichen-associated cyanobacteria. Over 2,000 strains we have at the moment. And surprisingly, we also found microcystins and nodularis in lichen-associated cyanobacteria. These cultures also showed us that the same species may have toxic and non-toxic strains. So in the microscope, we cannot differentiate toxic cyanobacteria from non-toxic ones. So at the beginning, people were saying that, are you sure that cyanobacteria are producers of these toxins and not other associated microbes? But when we got those strains axenic, we could really verify that they are produced by cyanobacteria. And now we think that these culture collections are a very valuable resource to study all kinds of aspects like structures of the toxins, bioactive compounds, molecular biology, taxonomic relationships, toxin biosynthesis, genomics, and many other things. Here is our culture collection person who takes care of Ludmila Saari. Here is the biosynthesis of microcystins in Anapena, which is nowadays also called Oligospermum. The taxonomy has been changing a lot during these years I have studied cyanobacteria. So you can see 55 kilobase long gene cluster is responsible for this microcystin synthesis. And there are different enzymes. These genes are coding for enzymes which are these balls represent polypeptide synthetases, and these squares represent non-ribosomal peptide synthetases. And the synthesis starts from the ADA amino acid, then each amino acid is added, and finally this thioesterase domain here makes this compound cyclic. So in microcystis and Planktothrix and Nostoc similar manner, these toxins are produced. So when we had revealed this biosynthesis of these toxins, we could also start to develop molecular tools to detect toxin-producing cyanobacteria. We designed primers in the conventional PCR reactions, and of course they are rather easy and cheap and allow analysis of large data set. For example, we studied over 70 lakes in Finland and see that often in Finnish lake at least several toxin producers occur in the same time. Then there is of course this quantitative real-time PCR method where you could see what is the main toxin producer. For example, we had a Lake Tuusulanjärvi where we noticed that at the beginning there was microcystis bloom dominating toxic bloom, and later also Anapena Oligospermum bloom became apparent. But every year we monitored that lake 2 years, and there was always these two producers present. Then we developed also this kind of chip assay where we could simultaneously detect toxin producers based on this microcystin synthetase probes, and then what cyanobacteria are present based on these 16S RNA probes. And we regarded it a very nice monitoring tool. Then there are also other methods like DGGE on MCYE genes and metagenomics, where also unknown diversity can be detected. And in the same cost action, we also made this kind of book, molecular tools for detection and quantification of toxigenic cyanobacteria. So that is available if you are interested. Here you can see phylogenetic tree of cyanobacteria. This is an old paper from 2013, and most of these red marked cultures, they came from Pasteur culture collection, and we get much more diversity. The dark colors indicate the genomes which were available, and you can see that this unicellular Synechococcus, which have very small genomes, plenty of those were earlier sequenced, the whole genomes. But then we decided to look for these biosynthetic gene clusters among these strains, and you can see that the blue color indicates polypeptide synthetase, green color non-ribosomal peptide synthetase, and these hybrids are marked with the red color. And you can see that this kind of biosynthetic system seems to be very common in these heteroclisters cyanobacteria, filamentous cyanobacteria, but not so common. Only polypeptide synthetase is in this unicellular cyanobacteria. But then we also, based on the genome, we could look at this bacteriocin, cyanobacteins, terpenes, and lantibiotics and others, and they seem to be, these kind of gene clusters seem to be very common in cyanobacteria. But what's more, we know only few compounds and biosynthetic enzymes, so there is plenty of research still to be done about these compounds and biosynthetic systems. So here is one example of our genome project. This Anapena 90, nowadays called Oligospermium 90, was isolated from Lake Vesijärvi, and you can notice that there is 2 chromosomes. At the beginning we thought that this is megaplasmid, but when it seemed to contain also genes, important genes for photosynthesis, we name it as a chromosome 2. And then we were interested to see where are these kind of biosynthetic gene clusters located. As you can see that they are all located in this biggest chromosome. There are these microcystin, anaboidopeptide, hassalidins. Hassalidins were the new finding from this genome, and we could later show that it's very common among other filamentous cyanobacteria. Then also these bacteriocin gene clusters were common, and anacyclamide, and altogether 250 kilo base pairs were dedicated for these biosynthetic systems. So what was also surprising finding was that cyanobacteria produce compounds which prevent apoptosis caused by cyanobacteria hepatotoxins. Here, if we incubate microcystins and nodularin together with the hepatocytes, here you can see what happens. There are these kind of systems where these microcystins and nodularin can enter in the hepatocytes and cause this apoptosis, cell death. But these compounds we found, they are also cyclic peptides, nostopeptides and nostopeptines, and they seem to be antitoxins. They blocked this transmission of nodularin, and that's why this apoptosis doesn't happen. So it was a kind of interesting finding. These hepatocytes were used as a detection method for hepatotoxins, but in this case, you can see that they might not be reliable always. So now I go to cyanobacteria blooms in the Baltic Sea. We know now that hepatotoxic blooms occur every summer. There is this nodularin, which is the Nodulares fumigena, which is producing nodularin and occurring in this open waters of the Baltic Sea. Here is Sweden, Gotland Island, Baltic countries, and Finland over here. And in the coastal areas, we have found also Anapena and Oligospermum, which are microcystin producers present in the Baltic Sea. So we decided to sequence some of the Baltic Sea strains. For example, here is results of phylogenetic tree based on these genomes. We selected 31 genes and made the tree, and you can see that this Dolicospermum from the Baltic Sea, which is here, was very similar to the Baltic Sea, which is the Dolicospermum species, which is coming from fresh waters. So, also, if you look at these Nodularia spumigena strains, 2 strains from the Baltic Sea, they were very, very similar, and one from Brazil, and it was also quite similar to Baltic Sea strains. So when we had these genomes, we decided to look for transcriptome and especially focused on phosphorus and salinity. And phosphorus, of course, because this bloom-forming cyanobacteria in the Baltic Sea, they are nitrogen fixers, and that's why the phosphorus is very important. And in salinity gradients varies in Baltic Sea very much, and that's why it was of interest. So I present these 3 papers in one slide. So first we are looking for Dolicospermum and phosphorus limitation, and you can see that when we were cultivating this strain in phosphorus limited conditions, it turned out to be shutting down this photosynthetic system. But on the other hand, you can see here green means induced genes and red color repressed genes. And what was more important was that this PST ABCS was very strongly upregulated, and this is genes related to phosphorus transport and assimilation. PHO regulon is very, very important, and that, of course, this organism tried to get inside the cells as much phosphorus as possible. Then here is the Dolicospermum and the high salinity. We noticed when we were cultivating this strain that it didn't stand high concentrations of saline, but even this 6 promille of salinity, it grew not so well. It started to be a little bit yellowish. You can see that again there is repression of photosynthesis, repression of nitrogen fixation because it's very expensive process, and then there is transcription and chaperones and lipids were genes were induced. So the remodeling of this cell wall definitely needed to do when there was a high salinity stress in this Dolicospermum. So here is Nodularia spumigena, and low salinity seems to be a stressful situation for this strain. We know that this Baltic Nodularia prefers to grow in a saline environment. And here you can see that everything again was repressed what comes to photosynthesis, but amino acid metabolism, transcription, and chaperones and translation were very active. So remodeling of the cell system was needed in order to this organism to survive this system. So my last story is about phosphonite degradation gene cluster in Cyanobacteria. We noticed that Cyanobacteria, these Nodularia strains, they contained this kind of PHN gene cluster, which is cutting the phosphorus carbon backbone here, and there is phosphorus liberated and also carbon compound. And we were looking also at other Cyanobacteria and found that 5% of all the sequenced genomes of Cyanobacteria were containing this kind of genes, phosphonate degrading genes. So it wasn't very common. And here you can see that all the 15 nodularis fumigaria cultures, they are having this gene, and then also in the Baltic Sea we could detect this gene in the blooms. So we think that cyanobacteria can use phosphonates and liberate methane. We were noticed that this methyl phosphonate was the one in which both of these strains were growing very well. And since they are photosynthetic organisms, they don't need this carbon, and that's why they liberate it in the water environment. So here are the conclusions. Mass occurrence of cyanobacteria are health risk for animals and humans, and it's a worldwide problem. Cyanobacteria produce various toxins and new bioactive compounds. Toxin biosynthesis allowed us to develop methods to detect toxic cyanobacteria. And of course, a combination of genomics, bioinformatics, biochemistry, bioactivity screening, and chemical identification and compounds are likely to yield a number of new discoveries. So thank you very much. And here are the persons I would like to thank as well.
Thank you very much, Kaarina. We have questions.
Over time. Sorry.
A little bit, but we have questions, so they're already here. I can read them to you.
Please do, yeah.
So the first comes from Eric Capo, and it says, are those compounds produced by cyanobacteria to prevent apoptosis also produced by other microbes, or they are only produced by the toxin-producing cyanobacteria?
We have studied only cyanobacteria and found surprisingly them and they are not very common. There are rather few reports about this kind of compounds occurring. So they are all coming from our laboratory. Nobody else has found them. And they are not very frequent among the cyanobacteria.
Thank you. Eric says thank you too. So the next question is from Malin Olofsson. How closely in time and space were the set of Anabaena dolichospermum and the set of Nodularia strains collected, the ones that you sequenced? How close then they were?
The other dolichospermum was sequenced from the lake, and the other one in the Gulf of Finland, the dolichospermum. And then these two Nodularia strains, the other one is coming from Gulf of Finland, and the other one is coming nearby Bornholm. So they are representing different areas.
Different areas. Thank you. And from Stefan Bertilsson, great talk. So it's known when in evolutionary terms cyanobacteria started to produce toxins. Is it something recent or an ancient trait?
We have studied that. I didn't show the results, but it seems to us that at least these microcysteine synthetase genes are ancient, and those strains which doesn't produce toxins, they have lost the genes. And we have seen also in the laboratory that sometimes we have a parts of the genes and not the full set of genes. So they are also in laboratory. They are losing those genes.
Okay, thank you so much.
Thank you for inviting me today. I was really happy that when we did this word diagram at the beginning of the meeting, that the first biggest word in the middle is drinking water. And I should confess that I didn't get mine to work, so I couldn't have biased the word map. So today I decided to talk about one study that we're working on, and it is entitled Long-Term Dynamics in Bacterial Biofilm Communities. So you see the words that I have there in highlighting are key. Living in drinking water pipes. So these are the pipes that bring the water to your house. And the roles that these biofilms have in nitrification and shaping the microbiome at the tap. And this is work that's been conducted by a number of partners, including myself, which you shall see if I can change the slide. There we go. Should be coming. No? Come on. There we go. Okay. So this is the core group that has participated in this study. And you see us in action at the top. So we have Sven Erik Ericson, Sandy Chan, Christian Pulleritz, Alexander Koikin, and Moshe Habigail. And on the bottom, we have Peter Rådström, Jon Allander, and Caroline Schleich. And the top picture was taken when we were sampling together with VIVAB, which is the drinking water company that we've been collaborating with from Lund University. And at the bottom, you see Jon Allander, and he is a bioinformatician at FOI, which is the Swedish Defense Research Agency. And Caroline works as a research engineer at VIVAB. So all of us together have been putting this data together. And when I first started to work on drinking water, I got really excited because I realized that somewhere between the lake and the laboratory, you have these drinking water treatment plants. And the people who work there, they see these as a factory to produce drinking water, but I see these as like really big replicates of all different kinds of microbiological mesocosms. So I'm just going to try the pointer and hope that that also works. There we go. So some of you may know this body of water. This is Mälaren outside of Stockholm. And here we have one of the waterworks that makes water for Stockholm. And you see a bunch of slow sand filters. And I'm not going to talk to you about these today, but we also work on the ecology of the bacteria living here that clean the water. Today, I'm going to talk about a project we have with a drinking water treatment plant where they installed ultrafiltration. And so to all of us, this looks like a machine. And to the drinking water treatment plant, this is removing a lot of organic matter, but also bacterial cells. And for the microbiologist, this is a really big filter, 0.22 microns, that removes a lot of bacteria. And what made me even more excited and happy is that the drinking water treatment plants, their job is to produce water 24 hours a day, 7 days a week for all of us. And that means that they collect a lot of data that describes all of these systems. So here we have Caroline showing her flow cytometer at VIVAB. So they actually count the cells on the outgoing water in real time. So this is an online flow cytometer. We have some ATP, I think this maybe is an ATP monitor. We have a sampling tap here to take water, and we have various other things measuring organic matter, pH, and yeah, and it's all collected. And then what should we do with all this information? So at least for me, I like to use it to look at the biofilm. So in drinking water, there's a lot of biofilm, as I'm sure you guys can imagine. Water means microbes and surfaces means biofilm. So we start in the source water. The source water comes into a treatment plant where you might have sand filters. There's Christian Pulleritz taking a sample, and there's Sandy Chan. This is actually making drinking water for Malmö, although it looks pretty green. We have biofiltration or oxidation steps during the treatment. We also have things like chlorination. There's the membrane filtration we're going to talk more about today, and ultraviolet light disinfection. And all of these are designed so that when the water heads out into the pipes on the way to your house, that water is going to stay safe. And the idea is that it should remain the same, biologically speaking, from when it leaves the treatment plant to when it arrives at the house. So we've also, there's Sandy sampling the pipes that I'm going to talk to you about today. And we also have had studies where we've looked at water meters, because this is the last biofilm. You can imagine there's a very, very tiny biofilm on those water meters just before the water heads into your house. And I can say that the biofilm is seen as a service because it acts to clean the water when it's inside the treatment plant, but it can also be seen as a threat depending on the location. So if we look closer down, if you imagine that the black is a pipe, you have the water flowing through here on the way to your house, and you have bacteria growing on the surface of the pipes. Some of these bacteria are participating in biocorrosion, depending on the material. You can also have, if it's a synthetic polymer pipe, you have material that comes from the pipe into the water and affects what's growing up here. You have hydraulic influences, you have cells coming off and going back on. And sometimes we also forget, but there are other things besides bacteria. So we also have free DNA, viruses, invertebrates, and protozoa. So here's the experiment. There's some pictures around the edge here. There's Sandy ready with her gloves and her ethanol to jump in there. Here's a pipe section being taken out of the distribution network. Measuring the temperature is really important to know that we have a stable system before we take our sample. And there's Moshe in action, having just taken a swab. And so what we have here is, this is a schematic of the drinking water treatment plant. The water comes in here. There's a filter with this just rapid sand. Then they have storage tanks, UV light, and then the water goes out into the pipes. And so what we were going to do is we were going to sample the water at different points. So that's these DP1s, sampling water, and also take sections of pipe and sample the biofilm from these pipes. So the flow goes in this area. So it's 1, 2, 3 in this direction, and biosample 4, biofilm sample 4. And the experiment was to look at all these different water and biofilm samples before and after the installation of ultrafiltration and coagulation. And what this process did in the treatment plant is it removed a lot of the organic matter, and it also removed almost all of the cells from the drinking water. So how much biofilm is in the drinking water? So we use flow cytometry to monitor the system. And we started out, here's before the ultrafilter in 2016, and it was about 700,000 cells per milliliter in the drinking water. And this varies across Sweden depending on where you live and depending on which treatments you have in your treatment plan. When the ultrafiltration started, we then started to see a drop, of course, in the number of cells. And the different measurements here are, this is the finished water. So this is the water just leaving the treatment plant. You see the diagram in the top right with the blue dot. And then as the water was flowing through the pipes, so there's the cells removed by coagulation, ultrafiltration. And then you see by the time it gets to this distribution point 1, you have this many cells. And then by DP2, you have this many. And by DP3, you have this many. And we could actually resolve this with the flow cytometer, because normally there's so many cells in these systems that the resolution, both of sequencing and flow cytometry, is compromised. But in this case, because the ultrafilter removed so many of the cells, we actually started to see how much biofilm was coming into the water. And this is a very close points to the treatment plant, so not allowing a lot of time or contact over the pipe. But if we look here, here's a map of Varberg, and we have about 500 or 50 kilometers distance here, and we see different counts. So we can see, for example, here we have a very high cell count because it's taken a long time to travel, and we have shorter traveling times with lower cell counts. And this was able to be correlated. We see the different sampling points here in an NMDS plot of flow cytometry profiles. And you can see that the contact area with the biofilm is correlated to the total cell count, but also the retention time in the pipe. So basically this means that the longer the pipe is in the water, the longer the water is in the pipe, the more bacteria you're going to have from biofilm. And we could actually show this with 16S rRNA amplicon sequencing. And this is source tracker analysis that was conducted by Jon Allander and Kristin Pulleritz on the samples taken from Varberg. And what we could see is that if you compared the water samples to biofilm and water libraries, before ultrafiltration, this is a proportion of the sequences in the amplicons, most of them are similar to the bacteria that were found in the raw water. However, after the start of ultrafiltration, you see that the orange bar gets bigger, and this shows the influence of the biofilm community on the water. And one of the biggest issues with ultrafiltration is that the cell counts are very low, and so we could actually see the KIT background coming through because of the low cell counts. So we could see that the identity of the biofilm bacteria became the identity of the water bacteria. And the big question, which we're still working on, is this a risk? Could UF, ultrafiltration, change who is in the biofilm? Would this change the risk? Or can it change who leaves the biofilm, which would also be condoned as risk? And this is largely based on the idea that biofilms can be reservoirs for pathogens, such as Legionella and Pseudomonas. So we see the surface here, and we see the deep biofilm. So this is the comparison of the 16S amplicons. Do I really only have 3 minutes, Anna? Oh my God. Okay. And we see the biofilm here, BF1, 2, and 3. And what we can see is that before ultrafiltration, we had certain groups of bacteria, and I'll tell you a little more about which groups later. And we see after ultrafiltration. So you see that there's a change, lots of green ones, then lots of brown ones, and that this is related to their position in the pipe going from upstream to downstream. And it's because of these groups. So the dark green ones are Nitrosamonidaceae, and they're actually metabolizing the disinfectant, which is in this case monochloramine. This monochloramine is converted to nitrite, which then as it flows downstream can be converted by the Nitrospiraceae and Hyphomicrobiaceae. But when you add the ultrafiltration, you remove a lot of organic matter. And what this does is it increases the concentration of ammonia that's more or less felt by the biofilm. And so we saw an increase in Nitrosamonidaceae after UF. We saw the Sphingemonidaceae disappearing. And we saw downstream also that we had Nitrospiraceae, as well as a little bit of Hyphomicrobiaceae blooming. And the interesting thing was that even though there were many fewer cells in the water, the chemical water quality was stable with respect to nitrogen compounds. So here you see the concentrations of ammonia, nitrite, and nitrate, and you see them when there were many cells in the water and also when there were few cells in the water. And what this means is that the only thing performing nitrogen conversion in the distribution system is the biofilm in the pipes. So even though there were 700,000 cells in the water per milliliter, nothing was affected when that was removed in terms of the nitrogen cycling. And it was also spatially distributed. Okay, I'm almost done. Here are the details. And the ASV level changes could explain what we saw. So if we look at the Nitrosamonidaceae upstream and downstream, we see that the amounts of one ASV are increasing in abundance, whereas another ASV is decreasing. And we also see this here. This is a lot of Nitrospira, 50% abundance, up to 75%, and reflected by changes in the other ASVs. We also see the same kind of trends when we look at the carbon cyclers. And with respect to risk, we can see, because that's what this is supposed to be, water safety, we see that the Legionella actually decrease. And we actually see the Mycobacterium increase. It's hard to see because of the scale, but it slightly increases. And at that point, I should say that, as most of you know, this is 16S amplicons, and we don't have the resolution to strain level. So this would require additional analysis. So to summarize, after ultrafiltration and coagulation, the water has many fewer cells, but not many other changes. We need to, from a risk perspective, consider which Mycobacterium and Legionella were detected. The ultrafiltration seems to have reduced the amount of Legionella in the biofilm, and the biofilm community has adapted to the ultrafiltered water. And I think this shows a little bit of functional redundancy in action, because the ASVs are tuning depending on the ammonium and nitrite concentrations. And then, of course, the big question is, what would actually happen if the chlorine was removed from the system? So that's a question we have as well. With that, I say thank you. There's a picture of the biofilm sitting on the pipe, that's BF1. And I can also extend an invitation to the PhD defense of Kristen Pulleritz. It will be online December 4th at 9 a.m. You can email me if you'd like a link. You can follow me at Twitter, or you can check out the Water Research School at Lund University. Thank you.
Thank you so much. Super interesting topic. And we have already a question. I also have a question, but I will start with the question that's here from Stefan Bertilsson. So he says, really interesting topic. What's the strategy for replicating your experiments? The impacts of ultrafiltration in this type of costly full-scale setting? Parallel pipes analyzed, or, for example, I guess not for stations linked by flow, as this would introduce dependencies between samples, or repeated experiments. So how could you replicate it?
I think the most direct way would be to try and recruit another treatment plant that would be interested in doing the same type of change in their treatment plant, and then follow what would happen in their biofilm as well. Of course, you could always, in the original study, you could always have tried other branches in the same distribution network, but it's very difficult and invasive to dig up actual pipes from the system. So this is why it's limited in that respect. So I think the best way to replicate would, to look at another system that's using monochloramine and was interested to use ultrafiltration.
Thank you. My question is about that you have all this nitrification with the nitrosomonas, and doesn't this increase the organic carbon? So these are like autotrophic microbes. Won't it increase the organic carbon and then like a cascade, providing more carbon source for?
Yeah, because I mean, the idea is that the microbes are going to multiply in the biofilm. And then, and it would have been really interesting to look even further downstream. Of course, you can always say that you can look more, because it would be interesting to see what would be even further down after the nitrosamonidaceae and the nitrospira. Would you get something else dominating the biofilm once the nitrite and nitrate had been absorbed? Sorry, nitrite had been absorbed.
Okay, thank you so much.
Yeah.
What I will talk about is how terrestrial dissolved organic matter influences bacterioplankton in the northern Baltic Sea, because I work mostly in the Baltic Sea. And the core group which have made this study is Daniela Figueroa, Owen Rowe, Joanna Paxkowska, and me. And then later in the project, Eric Capo made very great contribution to the project, which I will show you now. Then I share, do you see it? Yeah?
Yes, we can see it.
Okay, good. So according to climate change scenarios, of course, this will be a little bit of guessing, but this is sort of a summary of the present opinion and knowledge. So in the Baltic proper, like in the southern Baltic Sea, we can expect maybe more larger cyanobacterial blooms, what Kaarina Sivonen talked about. But in the northern Baltic Sea, like you see here, we can expect to get a more browner water due to heavier rainfall and if I click this, do you see myself? So because of here up in the northern Scandinavia, we can expect, according to climate research, we can expect more rainfall and increased runoff from land. So we will get a browning of the water and also lower salinity. So we will get higher color dissolved organic matter in the water. It's not really toxic itself, but it deteriorates the quality of the water, I would say, because it will make a poorer climate, a light climate, and decrease primary production and so on. While the heterotrophic bacteria may increase actually their activity. And this figure, I would like to show that already now in the Bothnian Sea, in the northern Baltic Sea, it's a really brown sea, let's say so.
Agneta,
Yeah?
someone says that if you click on the force icon below, then we will see it right.
If I click on...
No, well, if that, I think you can go back as it was.
Should I go back? Okay.
Yes, yes, yes.
Yeah.
Sorry, we won't disturb you.
Yeah, it's no problem.
We can see.
Anyway, let's say like today, maybe the food web starts here in the triangle with phytoplankton, and the energy is transferred up the food web in the rich fish. But in the future, maybe it will be more fed by bacteria and the primary production will decrease. So we get more trophic steps and lesser energy that reaches higher trophic levels. So we have made a study in which I would like to present to you in the northern Baltic Sea, in the Rhône estuary, which you can see here. Can you see my pointer? Yeah, hopefully. It's in the very north of the Baltic Sea. And we made a study during 2011 where we sampled 19 different stations in the estuary like this. And we studied 4 months, May, June, July, and August, and sampled 19 stations. And we measured many physicochemical variables and production measurements, and we also analyzed 16 sRNA gene amplicon sequencing. And you can see from the river discharge, it's very influenced by river discharge, this estuary. And you can see that when we did 4 samplings here, and the first sample in May was just at the end of the spring river flush. Then we had 3 additional samplings later in summer. And especially during spring, the estuary was really influenced by fresh water, as you can see from my evaluation of during different months in salinity. So the salinity was 0.2 PSUs in the estuary as a mean, while at an offshore station it's 3 PSU. So you can see it's really low salinity. And the influence of fresh water decreased during the summer. And the same we can see if we analyze dissolved organic carbon. Because the dissolved organic carbon is rather high in the river water and pretty low in the offshore station, and it's some sort of in the middle. But the message here is that in spring it's really influenced by fresh water, but this influence decrease. So it's a flow through flush of water, especially in spring. And then when we measure the function of the bacteria and primary producer, you can see that in difference to what it normally looks, it is high bacterial production in spring, and it decreased during the summer. It's the dark part of the bar, while the primary production actually show the opposite pattern. Quite unusual. And it's more like how it looks like in humic lakes than in clear water lakes or the marine system. So no spring bloom of phytoplankton, rather a bacterial maximum in spring. And this bacterial production follows rather much the concentration of dissolved organic carbon and humic substance, as you can see here. So even if the bioavailability of the DOC is low, the huge amount that flows through the estuary is really fueling. It looks like it's fueling the bacterial production. And regarding primary production, I said it's the opposite. It's the lowest, no spring bloom. It's increased during the summer, and it's really low. It's a phosphorus limited system, and the phosphorus concentration are low all through the season. And then we analyzed the bacterial composition. And if you look at here, the right part of the panel, you can see that in May, see here, the community, bacterial community is dominated by beta proteobacteria. This looks rather similar from the landward or the coastal stations and the more offshore stations. While in the summer, it's a more diverse system. You can see in August here that microbia is quite dominating, especially in the more offshore stations. So we have a temporal variation and a spatial variation also, especially in spatial variation, especially in summer. And then if we'll do a non-metric multidimensional scaling analysis, we can see that bacterial community composition is rather different in May compared to in summer, as you can see here, distributes different NMDS analysis. And, and also, if we put environmental factors, you can see that in May, for example, in all months, the landward or the coastal stations is like have a connection to DOC, high DOC concentrations coming from the river, humic substances, and color dissolved organic matter. And this kind of pattern you can sort of see in all months. And also when we relate the bacterial community structure to DOC, I mean the terrestrial components, we can see that DOC, humic substances, and CDOM has significant relationship to the bacterial community composition in during all 3 months, not only in spring, even stronger, more significant connection we can see later on. Anna has shown up here. And then we did a network analysis and for the different months, and we can see so each point is like a OTU, like the bacterial identity. And we can see that in May, there are lesser clusterings than during later part of the season. So we can say that in, each cluster is called a module then. So for example, here in July, we see strong clustering, and in May, very few, they were spread out. And we did, this is from statistical modeling from a method presented by Olesen et al. In 2007. And then it's possible to this model to sort of identify habitat specialist and habitat generalist. And the specialist is thought of have only few connections to each other, while the generalists have many links to each other, and they are in modules. And basically, I can, in simple words, the bacteria that ends up here, they are like specialists, and the ones that are in this part of the analysis, they are generalists. But anyway, in spring, we found that specialists are dominating, as you can see, while generalists are dominating during summer. And the modules, the clustered bacteria, when we did the Pearson correlation to, for example, terrestrial factors, we see that many modules have a positive relation to terrestrial compounds. So we think this is really something that affects the community structure. So my conclusion is that the terrestrial DOM, dissolved organic matter compounds, regulates bacterial community during the whole productive season. And that we found non-random association between bacterial populations, and this association different during different times of the year, but a recurrent cluster was beta-proteobacteria, Bacteroidetes, and actinobacteria. The functional roles that were identified by this network analysis found that that generalist bacteria emerged later during the summer, when the DUM were composed of both autochthonous DUM and terrestrial sources. While during the spring, when it was mostly terrestrial sources, it was another community that was specialist more on degrading DUM, it looks like. So, but since then, just a speculation, you could say that, or an outlook for the future, it may be that the consumption of terrestrial DUM in this kind of northerly coastal estuary will likely increase due to the selective forces for specialist bacteria. That could lead to, you know, a restructuring of the basal community, the lower trophic levels, the bacterial community, and possibly also to air-altered carbon and energy flows in the food web. So, and also I could say that regarding potentially pathogenic bacteria, we have a PhD student, Karin Ericson, who is probably listened to this, that she has found, especially in the low saline water, occurrence of Legionella, and also Rickettsia, Mycobacterium, and Pseudomonas. But I don't have data for it, but our preliminary data looks like that it's with lower salinity, we will get more of this kind of bacteria. The funding is from the Marine Strategic Research Environment, Ecochange, and Pharmas. And also I would like to thank Umeå Marine Sciences Center for making the sampling possible. Thank you.
We have a question from Matthias Hötzinger.
Okay.
And he asks, is there a big difference of water volume coming from the rivers in spring compared to summer?
Yes, it is. I think maybe, yeah, the flow rate is, as you can see from one of my figures, is much, much higher in spring. So like 10-fold higher or so than in summer, absolutely. So it is.
I have also a question about this, that you showed that there's much more specialists in the spring than later. Do you think this is because it's more like a soil water, which is usually characterized by more specialists?
Yeah, that is what I suspect. They are flushed out from the river, and they survive in the estuary, and they actually, previous studies have shown that with the increased little more salinity makes the DOC more available also and in the river water.
There is one more question from Johan Wickner. Do you know what taxa that were the specialists and what specific catabolic properties they had?
Let me say that sometimes the specialists were the same all through the season, but in other cases, they switched from being specialist to generalist. So it was not constant. I cannot say that one taxa belonged to a specialist all the time, but this is how this statistical analysis turned out.
That's really interesting.
Yeah, and I don't remember exactly. Yeah, it's different annotations. Yeah, so I cannot recall exactly.
Okay, so I think anyone? Yeah, we stop here. There can be still questions in this Q&A to Agneta, but we will have also the breakout rooms. So now we have a leg stretcher.
I am ready.
Okay, then we will continue with Karin Rengefors. And I am really amazed by the title. I know your work about browning and phytoplankton, but as I see now, we will hear something different about how you move to water quality from fundamental science. So the floor is yours. I'll let you talk, and then my face will come up 3 minutes before the end. Thank you.
All right, thank you very much for that kind introduction. And yes, I wanted to do something a little bit different today, and I will talk a little bit about results from the research in our group, but I also wanted to talk about the research process and how unexpected findings can lead you down new paths. But first of all, let's move into the phytoplankton world. Just hold on here. I'm going to get my cursor in the right position. So, and specifically freshwater cyanobacteria. And as you all know, phytoplankton are at the base of the food web and are really important for the whole ecosystem functioning. And you also know that they affect water quality. And I think both Karin and Kaarina set the stage very well for me. And when it comes to freshwater phytoplankton, the cyanobacteria are usually a big and important component. But not only that, they also cause blooms in lakes. And the problem with these blooms, as you've heard before, is that many species produce toxins. And the most common of those toxins is microcystin that Kaarina introduced to us. And it's a hepatotoxin, so it causes liver damage. And of course, it's dangerous then for humans and dogs and cattle and horses to drink and can lead to death, worst case scenario. Now, the special problem about freshwater, in contrast to marine water, is that we actually do drink this water. And in Sweden, actually 50% of all the drinking water is from surface waters. And in our case, that means mostly natural lakes and not even reservoirs. And so because of this, a lot of research has been done on toxic cyanobacterial blooms. And it was explained to us before. We know there are several different species. This is microcystis is one of them. The toxin has been characterized. But even though all this excellent research has been done, there's still some unanswered questions. And one of them is that we're really poor at predicting when blooms will become toxic. That is, we don't know always when in a season, or not even if one particular lake will be get toxic blooms or not. And part of this is that some of the strains within the species do not produce any toxins at all. And so the question is also, so why do we have both toxin producing and non-toxin producing strains, and how can those coexist? The third question is, or the poor understanding is actually why they produced microcystins. So there have been a number of hypotheses and ideas, but it's still not clear why and why there are so many variants. And to my understanding, there are at least 240 variants of microcystins that have been described, and there will probably be more coming in the future. So what my research group did, we became interested in the cyanobacteria and left the eukaryotes for a while and looked into a species called Microcystis botrys, because this species is very common in the south of Sweden and it forms toxic blooms. And there are three fundamental research questions we're asking about these blooms. And the first one is, do toxin and non-toxin producing strains form separate subpopulations? That is, are they really genetically different in more ways than in the toxin production? And the second question is, what are the population genomic differences? So looking at the whole genome, what are the difference between toxin and non-toxin producing strains? And finally, what are the different factors that favor one type of strain over the other? And this work is being performed by Emma Johansson, who is a PhD student, and I think she is listening in here. And here I will present Emma's first study, which was on the diversity of microcystin chemotypes within the summer bloom of Microcystis botrys. And the objectives of this study was to investigate the temporal dynamics of microcystin and non-microcystin producing strains of Microcystis botrys, and to check what kind of microcystin variants were produced. And so to do this, we worked in Lake Vombsjön. Lake Vombsjön is a drinking water supply to Malmö. And we sampled this across the summer, 5 different occasions, isolated single colonies, established cultures, and then harvested the cultures and determined their microcystin profiles by mass spectroscopy. And so what did Emma find? Well, one of the major findings was that only 28% of those strains actually produce microcystins, meaning that the majority actually did not produce any microcystins at all. And moreover, she found that those that produced the microcystins also were for the most part unique. So they produced a unique combination of microcystins. So let's have a look at what that looked like. So if we look at this table, you have all the strains here in the first column, and the S1 means sampling 1 and so forth. And then across the top, you have the different variants. Don't worry so much about what they're called, but look for the black dots. The black dots signify different kinds of microcystin variants. And so if we look a bit more in detail of this, you can see that some strains have very many dots, and like these down here, and then some have only a few variants. So there is a difference between the types. And moreover, there seems to be seasonal difference. So the strains in the beginning of the summer have a lot of variants, and so do the ones at the end of the summer, but not so much the ones in the middle. Now, in addition to that, microcystin producing strains dominated towards the end of the summer. So this figure shows the proportion of microcystin producing to non-producing strains, and the red ones are the microcystin producers. And you can see they're quite only a low percentage in the beginning of the summer, but then in August, September, there's a bigger proportion of the toxin producers. And so the question is, and this is statistically significant, and the question is, well, what's happening here? Well, we think it may have something to do with nutrient quotas, because the quota between nitrogen to phosphorus dropped down to about 8 at the end of the summer, meaning that this lake switched from being phosphorus limited to being nitrogen limited, and this coincided with an increased proportion of toxic strains. So possibly there is an answer somewhere there when looking at the nutrient quotas. And so because of that, Emma carried on a more extensive field study looking into the strain dynamics and the environmental parameters. And she did this in the summer of 2018 and 2019. And here I will switch gears a little bit and talk about curiosity. And so one of the things I do, which is very, very nerdy, but I tend to carry along a plankton net when I travel or I'm out for the day. And I take a plankton sample in a pond or a lake that I encounter. And sometimes you find nothing interesting, but other times you do. So what happened in the summer of 2018 is that we expected a huge cyanobacterial bloom. And then Emma said when she did the sampling, well, it doesn't seem like there's much cyanobacteria. There are other things in there, flagellates and other non-cyanobacteria. And so, of course, I was curious. When I was out one day taking my dad for a little tour around the countryside, we stopped, took a plankton sample. I brought it back to the lab, and lo and behold, there was a dinoflagellate. And I was super excited because I love dinoflagellates and did my PhD on dinoflagellates. And so I isolated cells, I grew up cultures just for the fun of it, but also to be able to determine which species it was. And it turned out that it's a species called Neodymium polonicum. And this is very interesting because the species is quite rare. However, it is also the only known freshwater dinoflagellate to produce toxins, and it produces a toxin called polonitoxin. And so I was delighted. I thought this was very cool. And then I wanted to look at the data. And so when we got the phytoplankton data, what did we see? Well, first here, I'm showing you a regular year, which turns out to be 2019. But the typical thing you see in the phytoplankton composition, which this shows, is that the cyanobacteria, which are the turquoise, tend to dominate completely in the summer. And don't worry about the other colors, but the gray are the dinoflagellates, so you can pay attention to those. So what did 2018 look like then? Well, here we were so surprised, but it was the reverse. The dinoflagellates dominated already in the beginning of the summer and all through the summer season. So this summer of 2018 was very unusual in terms of the phytoplankton community. And as you know, it's also a summer that was extremely hot and dry, and many degrees over the regular temperatures. And so I was thinking, it would be so exciting to study this dinoflagellate, but we don't have money for that project. But I start to think about why could this be? And then very lucky, it turned out there was an opportunity in the fall of 2018, and the Research Council Formas made an announcement that they would have a call on extreme weather, and you needed to have preliminary data to be able to apply to this call. And of course, I had already thought of the idea, and I was so lucky. I wrote the proposal, called it Blooms Like It Hot or Not, and the proposal was approved and granted. Now, the interesting thing with these proposals is that they wanted a societal connection. And so the obvious connection here, of course, was the drinking water supply, Lake Vomsjön, where we had seen the dinoflagellate bloom. And this is, as I said, drinking water to Malmö, and it supplies 500,000 people with drinking water. So it's quite important. But surprisingly, there's no monitoring of phytoplankton and no monitoring of microsystems in this lake. And so here I saw my chance, and we thought that it would be really nice to have a citizen science project to approach this monitoring. And that's why we started the citizen science project Algväktarna, which means something like algae watchers. And I want to say here that this project was inspired by a project at the Linnaeus University many years ago called Algspanarna, and they helped me a lot to get started and give me ideas of what to do. Anyhow, the aim here was to follow the temporal and spatial dynamics of algal blooms in Lake Vomsjön, and at the same time engage the public. And the way we planned this is we started by contacting potential volunteers by direct mail and email to people who lived around the lake, also by announcements on websites and newspapers. Then we allowed the volunteers to register their interest. And in the meantime, in the lab, we developed the methods and the methods handbook and the methods video. And then eventually at the beginning of the summer, we had prepared those methods and trained the algae watchers. And what they actually did then is that they sampled daily samples, 5 locations, at the lake. They sampled surface water, which was then filtered down onto a filter. They compared the filters against a color chart that we had prepared, and each color corresponded to biomass that we had calculated in the lab. And then they reported back to us on their phones on a form. And when we received their reports, we reported back to the public on a public website. And here the public could enter and check the water quality for swimming and also microcystin levels and so forth. So the outcomes of this, well, first of all, I must say the unexpected media attention. We had TV, we had radio, we had newspapers, first page on the newspapers, and that was of course so exciting for all the volunteers who were doing all the hard work. And in addition, the volunteers collected a total of 454 samples across 90 days, which I think is absolutely amazing. And looking forward now to analyzing that data more in depth. And with that, I would like to acknowledge everyone who was involved in these projects, the funding agencies, and I would like to thank you for listening. Thank you very much.
Thank you so much, Karin. Really amazing presentation and really cool project. We have questions and we have time for questions. So I want to encourage everyone. I wanted to mention that we have 70 participants in this webinar. So please go ahead. So we have a question from Stefan, but I will start with another one. So Tiancho Su asks, can one strain of cyanobacteria switch on-off toxin production under different environmental conditions?
So what we've seen from our lab strains is that they either have the microcystin gene and they produce the toxins, or when they don't produce the toxins, they lack the gene. And I think Kaarina knows much more about this. And I understand that to some extent they can modify their production, but as far as I know, they don't close it down completely unless they have lost the genes or there's a mutation.
So they can turn it off, basically. It's different.
That's my understanding, that they don't turn it off. But Kaarina can correct me if I'm wrong here.
I fully agree with you. Yeah, that's the case in our according to our research as well.
Then we have, I will read. Yeah, go ahead.
Could I just follow on the same question? And I'm sorry to the people in the chat, the panel, we just have to turn on our camera and jump right in for a question. But I was curious whether the toxin production would be different if you culture in the lab or if it would be in the lake. And that kind of follows on Sanchio's question. And now I'll turn my camera.
Absolutely. Of course, in the one thing we could do in the lab is that we could culture everything under the same conditions, but it could probably be modified in the field. And I think that's also been shown, that if you change the environmental conditions, you can regulate the toxin production somewhat, but you don't turn it on or off. But what we do see in the lab, just like Kaarina, is that they lose their genes. Under, I don't know which circumstances, but it happens.
Then I will proceed with the questions. So from Stefan, hi Karin, are the microcystin variant profiles for the different strains stable under different environmental conditions? This is quite similar, I think. Have you planned or done some more quantitative assessment of the abundance of the different strains with different toxin profiles over the year?
So this is Emma's project for 2018 and 19. So she's in the middle of analyzing those samples with qPCR. So we will have to get back to you on those, and with the ELISAs for the microcystin in the water.
Hey, I also comment. It was interesting to see what toxins were most common in the lake. So we have seen similar thing that this microcystin RR and demethylated microcystins and microcystin LR are the most common toxins also in Finnish lakes. So it was diversity.
Very much. We need to compare some notes, I think.
Then we have more toxin questions. I will go with those first. One from Rhiannon Mondav from Uppsala. So to both of you, Kaarina and Karin. So is microcystin ribosomally or non-ribosomally synthesized?
It's
non-ribosomally. As I showed in my presentation about the in Doligospermum, in microcystis is the same, that there are this hybrid polyketide non-ribosomal peptide synthetases which are creating the compound.
Wow, that's really amazing. And one from Anders Andersson. Did the toxins from dinoflagellates cause problems with the drinking water in 2018?
We don't think so. So the thing is with that Neodymium polonicum, nobody measured the toxins. We didn't even know about it until I had cultured those cells. So and it requires special methodology to measure that. But at least they are known to be ichthyotoxic, so toxic against fish. But there were no reports of fish kills. So it might be that specific population is not a toxin producer, but we don't know that yet. Okay, thank you. And now quickly we have a lot of questions about citizen science projects. So I will just ask you them together. So one is, how can you apply for it, from Ying Li? One from Elena Gorkova. How did you get from the filter colors reported by the volunteers to the toxin concentrations? And there is one more about if you will continue with the citizen science sampling efforts. All right, so the funding came from Formas, so that was part of the project, and Formas kind of required some, not citizen science, but some kind of societal connection. And then the second question, will we continue? We will see. We will have a closure meeting with all the algae watchers, and we'll see if they're interested in continuing next year, because it depends mostly on them. There is one more question. And the toxins, yeah. So, to this one, I forgot, how much training did they get? A 2-hour training. But then, of course, we've been in touch with them all the time via a Slack site. And so there's always a lot of feedback. And then, of course, we had to upload the data every day, even Saturday and Sunday, of course. So there's a lot of work all through this period. As to answer the toxins, and I didn't mention this during the talk, for the toxins, we use these dipsticks to measure microcystins, and so this was not monitored all the time. We tested this with some of the algae watchers. This was more difficult for them, and we didn't do it as frequently because it's much more expensive. But we've tried it out, and so if we continue next year, I think we will train some of the algae watchers to maybe only do the microcystins. Thank you very much. Now we have to continue with our next presenter. Thank you so much, Karin, for the presentation. And now the next speaker is Hélene Norder.
Yes. Hello.
Hi, Hélene. Hélene is a professor in virology, so we are jumping to much smaller organisms, or if we can, I think we can call them organisms, maybe not living ones. And we are jumping to wastewater, and I'm especially interested in this presentation of you. Hélene joins us from Gothenburg. She's a professor at Gothenburg University, and her presentation is about what happens through 1 year to viruses in wastewater.
Okay, so yes, I want to tell you something about the viruses in wastewater that we followed for 1 year. That was in 2017. And we followed it in the Ryaverken Gryab. And during that year, Gryab received about 130 millions of cubic square wastewater. So nearly 500 liters wastewater per person a day. And during the rainy state was much more. And we sampled every second week the incoming wastewater, and monthly we sampled the outgoing wastewater after the treatment that they do in Gryab. And we sampled according to the flow. As you know, when it's raining, they get much more wastewater in, whether in low raining days, there are much less. So to be sure that we get just as much from each toilet, so to say, because we wanted to see how much viruses there were in the society, we took according to the flow. So 30 milliliters were sampled per 1,000 times up to 3 liters of water during 24 hours. And nearly 2% of that daily samples were pooled for each day until it became 1 weekly sample. And that weekly sample we analyzed for viruses. So some week we could have 1 liter, and some weeks we could have 5 liters, depending on the flow. And we concentrated the wastewater that was incoming by absorption of the viruses into milk powder. But for the effluent, since it was really purified, we connected a filter, charge filter, and we let 10,000 liters of treated wastewater pass through the filter during 24 hours, 8 liters per minute. And then we eluted the viruses from the filter. And we did, when we had eluted and extracted the nucleic acid, we made qPCR to quantify 13 different viruses. And the amount of viruses were compared with the number of patients diagnosed with the virus during the same time period, and patients that were living in Gothenburg area. And for the effluent, we also used NGS with random primers to see if we could see more viruses than we could find by the qPCR. And what we found was that there were different patterns of these viruses. So for astrovirus, that gives more diseases in children and elderly, there were higher amounts. So this is a log scale. So higher amounts during the spring, and then we also have more patients infected. And then when there were less patients, there were less virus. And the same goes for the rotavirus. And for adeno, it peaked just some few weeks before it peaked in patients. And for noro-G1, which does not give really disease, it was an even very high amount all the year through. As for G2, that gives the winter vomiting disease, it was high during the time we had the patients, and then it went down during the summer, and then it peaked before the patients came. So a few weeks before the patients really entered into the hospital or to the health system, there were more viruses in the sewage. So we could really say in a few weeks, we will have more patients. And for G4, that doesn't really give any disease, or very few gets ill. It was rather even, although it fluctuated as well. For the other viruses, as sapovirus, which is also a calicivirus like norovirus, it was the same pattern. When we had a lot of patients, we had a high level in the sewage, in the incoming sewage, and then it decreased, and then it increased before we saw the patients again. For enteroviruses, you know, these viruses give aseptic meningitis often, and years when we have a lot of enterovirus outbreaks, it really increases here during the autumn. But 2017 was not an enterovirus year, and it was really lowered instead of increasing during that period, when it usually goes up. And then there were two other viruses that we do really not diagnose. One is parechoviruses, and we were surprised to find that much of parechovirus in the sewage. And this virus gives severe disease in small babies less than 1 year of age. That can go neurological disease, severe neurological disease. And for Aichi virus, it can also give gastrointestinal diseases. And we don't really investigate for that at the hospital. But we do have really high amounts, 10 to the 6 in the incoming. So it's probably there, but we should add it into our diagnosis. And also to our surprise, we found a lot of hepatitis A virus, although we had very few patients in Gothenburg during this year, only about 16 patients, but every week we did find virus there, which means that there are people with hepatitis A, and they really don't go to the hospital or they don't feel ill. They also have subclinical disease. And we also then checked in the effluents to see if we find hepatitis A there, and we did. So it was really there. And when we looked for the effluents, we could see that for each of those that came in, we also found them in the outgoing effluents, but with a 10,000 reductions. So for 10 logs, it was for all of them. And to our surprise, we also find hepatitis E virus in the effluent, but not in the influence. We were a bit surprised about that and wondered, why didn't we saw it here? Because it has the same amount that we see for enteroviruses or for Aichi and Salpy. So we did amplify the genes we had here by ordinary PCR and sequencing to see which strains there were. And we found 7 different strains. And most of them were strains with the genotype 3CI. And that genotype is really causing chronic hepatitis C. Not everyone that gets it gets chronic hepatitis C, but those that get chronic hepatitis C usually are infected by this strain. And we don't find this virus in Swedish pigs or in Swedish wild boars. But we do find it, the blue ones here are Swedish humans that are infected. We do find it here, and we see it in German pigs and in German wild boars. And here we have the red ones are the ones we found in the effluents. And then we also found the 3F. That is the strain we find in Swedish wild boars and in Swedish pigs, and also in Swedish acute cases. But in addition to these two, we also found 2 different strains, and they were very similar to hepatitis E strains infecting rats. And the genotype of these strains were really divergent. It was between the strains infecting European rats and those infecting Asian rats. And we know that those infecting Asian rats, they infect humans also, but we don't know whether these strains also can infect humans. We are going to look more careful for that. And we also found, of course, a lot of other viruses in the effluents. About up to 16% of all the reads we had when we made NGS were viral reads that we could classify as viral reads. And most of them were bacteriophages, and some very few were plant viruses, and some were circoviruses, which also we know infect birds and also humans and other animals. So there were also the mimiviruses that infect algae and are huge. They are as large as bacteria, close to. So there were a lot of viruses also, but we didn't find any other as we could define as human pathogens apart from those that we are checked by qPCR. And this year, when we had this COVID outbreak, we continued, or we restarted really, to look if we could find SARS-CoV-2 in the sewage or not. And we started week 7, and in week 14, we started also to check for the outgoing, the effluent, to see if it was incoming and effluent. And you can see here, the blue lines are the incoming amount of SARS-CoV-2 in the influence, in the sewage, and the red line is the effluent sewage. And we see that when this peaks in the incoming, it also peaks in the effluents. And it really followed and could really confirm what we saw in the influence. And again, it was about 4 logs 10,000 times reduction for the outgoing than for the incoming, but we don't know whether these are viable or not. That is what we are going to work on now. And when we compared, we started in week 7, which was the week that we had this winter holiday, school winter holiday in Gothenburg. And the blue line is the incoming. And then in week 8, the week after, we found some in the wastewater incoming, and then it disappeared, and it started to come back in week 11. And then we used the week 11 as a reference for the amount. And you can see in week 14, we had a peak. In week 19, we had a peak, and 22, and in 26. And we compared these peaks with those with the newly hospitalized patients with COVID. We could see that they were about 19 days from the peak in the wastewater until there was a peak in newly hospitalized patients. See, for 19, you have it here, you have it here. We didn't see that when we come into June, July, end of June and the beginning of July. Then it was not really a peak of newly hospitalized patients. And here, my grant ended, so I couldn't continue, unfortunately. So another thing that happened during this outbreak was that we looked at the other viruses at the same time. And what we saw was that all these fecal oral spread viruses, especially the norovirus 2, which usually peaks up here in week 11, they all declined in number, which means that the recommendations that we got to wash our hands, keep physical distance, and so on, made that all these viruses declined rapidly in the society as well as when we didn't see any outbreaks at all of any of them. So by identifying viruses in wastewater, we can use it to have it as a sentinel for outbreaks. We can use that tool to investigate the reduction of viruses in a wastewater treatment plant. We know it's about 4 logs. And we can use it as a tool to identify new viruses with possible pathogenicity for humans or animals. And I just want to acknowledge my colleagues that was working with this, Hao Wang, Freddie, and Ellen, that have done all this huge work. So thank you.
Thank you so much. Very interesting presentation of on a very hot topic right now. And we have time for questions. We will have the breakout rooms very soon, but I think we have time for 2 questions. There is one here from Moritz Bach. How specific are the primers for the viruses, mostly the less known ones? Have they been computationally tested against wastewater metagenomes or transcriptomes?
They are, the qPCR primers are very specialized for each virus. We use it in the routine. So, I mean, we know for sure that what we detect, that is the virus we detect. And they are so divergent, all the viruses, that it's really difficult to find them all. So I can assure you that they are very specific.
Thank you. Then there are a lot more questions. Emily Nielsen asks, thank you for a nice talk. I might have missed it, but why do you think that you saw hepatitis E in the effluent and not the influent?
I saw it by the metagenomics, by NGS. I identified hepatitis E virus, but we didn't see it in the influent when we used the qPCR. So we had to go back, and when we tested in qPCR in the metagenomics, then we saw the hepatitis E virus, and then we started to sequence in the effluent. We couldn't really identify it in the influent for some reason that we don't know why. Perhaps we had more of the rat hepatitis E. That doesn't match with the primers.
I was also thinking, can that hepatitis come from rats in the system?
Yes, yes, it can.
Thank you. One more question from Stefan Bertilsson. Is all the water arriving to the wastewater system from households, or is there also hospitals, farms, or anything livestock connected?
There's no livestock, no slaughtering, but there's a hospital is connected also. The Gothenburg Hospital, Sahlgrenska University Hospital is connected. Otherwise, it's households and, I mean, rainy water, daily water like that.
Yeah, and the other question you partly answered it. Does the number of people producing the wastewater differ over the year?
It's all the households of Gothenburg, over 790,000 inhabitants that are, not all are connected to, but close to all are connected to the rehab.
Yeah, I think that's it. There is one more, but that's not a question so much. That, but maybe, why is COVID-19 load so variable during the spring in the wastewater? What do you think?
Well, I think we have cluster spreads. It's not variable. It's variable all the time. It goes up and it goes down because we have spread in certain clusters. And when that cluster is spreading, then it goes up. And when it's spread enough, and then we get a new cluster somewhere else in the city, and then we get it up again. So I think that's the way it's spreading. Still is.
Mm-hmm. Thank you very much.
And it is my honor and privilege to introduce our next speaker for the scientific program, Professor Rita Caldwell, the distinguished professor from both University of Maryland and Johns Hopkins Bloomberg School of Public Health. Rita, you are internationally recognized for your research and prevention of waterborne diseases. Your interdisciplinary and formal backgrounds in bacteriology, genetics, and oceanography have shaped and complemented your research. From your nearly 600 publications, one can see that in your work, you have used the environment as a means to inform, predict, and prevent the spread of disease. You used your expertise and ingenuity to bring you into the entrepreneurial world, founding Cosmos ID in 2008, a bioinformatics company dedicated to improving global health by developing analytics for studying microbial activity. I see that Cosmos ID responded rather immediately to the current pandemic by providing several analytical services, including wastewater analysis, stool testing, and targeted genome sequencing of SARS coronavirus 2. Last we met a few years ago, we discussed why you chose bacteriology, and your response was, there weren't too many choices for women interested in sciences back then. Perhaps this was the motivation for your later administrative journey as the first female National Science Foundation director in the late 90s. During that time, you worked as a strong proponent of women in science, doubling the advanced budget, an initiative set up to increase opportunities for women in the fields of science and engineering in the United States. I personally know several female researchers in the field that have directly benefited from the advanced program. You've recently written a book entitled A Lab of One's Own, One Woman's Personal Journey Through Sexism in Science. We are honored to have you here with us today as a pioneer in the field and also as a strong role model and voice for women in science. Welcome.
Thank you very much, Rachel, for the very kind introduction, and Caroline for all the coordination to fit into my schedule. This is a kind of a crazy week. Well, 2020 is a crazy year. I would like to describe a bit of previous work and tie it into our current work on COVID-19. So if I may go through the slides quickly, I'm having you show them just to make sure we don't have any technical hiccups. So for the next slide, please. You're all focused in your studies on water, as I have been my career, mainly because diseases transmitted by water are in the billions of cases and millions of deaths per year. I particularly focused on diarrheal diseases, namely cholera, which has about 1.5 billion cases per year and at least 2 million deaths. As you know from the COVID-19 epidemic, we're certainly approaching the level of the waterborne diseases, though it is a virus, as opposed to the diarrheal diseases, which are predominantly bacterial. We have the next slide, please. Next, yes. I'll go through these quickly. Cholera is an acute water-related diarrheal disease. Next slide. It is occurring in pandemics, and the 7th pandemic that's been recorded began in the 1960s and continues today. Next slide. Cholera occurs today in more than 50 countries. It's really a pandemic. It affects at least 7 million people, some severely, resulting in death, others just mildly ill. Next slide, please. Unfairly, we ascribe the Bengal Delta as the homeland of cholera outbreaks, but 100 years ago, 150 years ago, Sweden, Scandinavia, US, coastal cities, inland cities, all suffered cholera on a regular basis during the summer months, during the warm months of the year. May I have the next slide, please? Cholera is really an aquatic bacterium, the Vibrio cholerae. Therefore, it's an aquatic transmitted disease. Now, we showed about 25 years ago that Vibrio cholerae is an aquatic bacterium. It was, that notion or hypothesis was resisted very strongly by the medical community. We have the next slide, please. However, it's now in the textbooks, and we have new biotypes emerging, which is again proof that these biotypes from the environment, the 0139 a dozen years ago, emerged, and it's an epidemic form of cholera. So the cholera bacteria carry out functions such as carbon cycling, nitrogen fixation, etc. At least 20% of the strains that I've tested are bioluminescent, and they all, Vibrios, produce a chitinolytic enzyme, breaking down the N-acetylglucosamine polymer that forms the basis of the structure of shrimp and crabs and other crustaceans. So its native habitat is the aquatic environment, and it is an aquatic bacterium. Have the next slide, please. So we were able to, next slide. So in our studies in the Chesapeake Bay, I started out my research at the University of Washington, and I elaborated in my book the tortured path that I had to take 50 or 60 years ago to get an education, because women were not really welcome in science. We weren't considered to be appropriate students of science. But nevertheless, I pursued my ambitions, and I achieved my goals eventually by being the first marine microbiologist to be trained as such in the United States at the University of Washington, postdoc at the National Research Council in Canada with Dr. Norman Gibbons, who was working on halophiles, and then took a position at the university, at Georgetown University in Maryland. While at Georgetown, I connected with the Johns Hopkins University, particularly their Chesapeake Bay laboratory, where the Ridgely Warfield was a catamaran that we used to traverse the bay. And you can see the stations numbered throughout the Chesapeake Bay in the United States. In the Mid-Bay region, station 834 and 818 was really where we picked up Vibrio cholerae, essentially from Kent Island down to the Horn Point Laboratory, where the salinity is about 15 parts per thousand. And so it's semi-saline. Down in the mouth of the bay, Vibrio parahemolyticus was dominant. We were the first to isolate the organism outside of Japan. And of course now Vibrio parahemolyticus, a foodborne pathogen, is distributed throughout the world. Next slide, please. The discoveries that we made included showing that the Vibrio, as you can see in the slide, Vibrio cholerae, a curved bacterium with a polar flagellum. We showed that its host is the copepod, which is a microscopic crustacean, I guess you would say, which is dominant in estuarine, riverine systems, and even in freshwater systems, various species of the copepod. We showed this back in the 1970s, work of a former student, Dr. Tatsuo Kaneko. We have the next slide, please. This is the copepod. The egg sac carries the, you can see to the left, carries the eggs of this gravid female about to release her eggs into the water column. The Vibrios coat the egg case, and they also, with their proteolytic enzyme, very likely are either commensal or symbiotic in opening the egg case and allowing the eggs to be distributed into the water column. So the relationship of the Vibrio to the copepod very likely is symbiotic, has not been shown as such, but definitely commensal for sure. May I have the next slide, please? We developed an early model from the Chesapeake Bay work that during the spring months when there's abundant sunshine, phytoplankton blooms occur. Then they're followed by the zooplankton blooms, which feed on the phytoplankton, the grass of the sea. And when the zooplankton bloom crashes, the Vibrios are released into the water column. And those who drink the water without filtration or treatment can ingest the Vibrios. And it's been shown by Richard Cash and his colleagues about 50 years ago that Vibrio cholerae is a dose-dependent disease. You need about 1 million cells per milliliter per teaspoon of water, so to speak, in order to come down with a full-blown case of cholera. If you ingest a few cells, you might have a mild diarrhea or vomiting, but not necessarily lethal. So this is the general model, very crude, that we developed way back in the work in the 70s in Chesapeake Bay. May I have the next slide, please? I was invited to Bangladesh because we had published a good deal of our work to see whether our model really held in a cholera ridden country like Bangladesh. And so indeed, we did our work there, next slide. And it was obvious from the ponds which the houses surrounded, where women mainly collected water for the household, did the washing of food for the family meals, washing utensils. It was also used for personal washing, toothbrushing, and in the far corner is the drainage of a latrine into the pond around which the houses are located. Quite obviously, the bacteria don't behave and stay in their corner, and therefore transmission is clearly person to person and from the environment. May I have the next slide, please? So from the work in Bangladesh, it was very clear back in the 70s and 80s that what we showed in the Chesapeake Bay held for Bangladesh as well. The bacteria are present in the water associated with copepods in Bangladesh, particularly in the Sundarbans, where along the coast of the Bay of Bengal. Next slide. Obviously, person-to-person transmission, next slide, occurs, but next slide, it's really the environment that's the source of the bacteria. So we confirmed the studies done in the United States in the cholera endemic country of Bangladesh. May I have the next slide, please? While working in Bangladesh, it occurred to me that, now this is some 25, 30 years ago, that the Landsat satellite that had just been launched might be able to be used to predict cholera outbreaks. The Landsat satellite had sensors that measured chlorophyll, sea surface temperature, sea surface height. Now, we had shown the parameters of salinity and temperature, a variety of nutrients, variety of other environmental parameters associated with the onset of cholera. We have the next slide. So we tried out a study where we measured the Bay of Bengal, a quadrant right off the immediate coast in the Sundarbans coastal area of Bangladesh, and the cholera cases in MATLAB throughout the year. And we were able to show a very striking correlation with, of course, a correction in the sense that the red lines are the cholera cases and the sea surface temperature is the blue lines. And the cholera cases, if corrected by correcting by 6 weeks forward, the correlation was strikingly strong. And mind you, this is a publication 20 years ago in PNAS without much of a sophisticated model, a very simple simplistic model, but yet we were able to show that indeed satellite imagery could be a powerful tool for public health. May I have the next slide, please? We were able to demonstrate for Kolkata, and Madla, Bangladesh, next slide, that the correlation between cholera and chlorophyll was powerful. A milligram per cubic meter of chlorophyll increase in concentration measured by satellite result was correlated with a third increase in the number of cholera cases in Calcutta. And from our other ground truth data, a millimeter per day of increase in rainfall resulted or was correlated with a 7% increase in cholera cases. And next slide, similarly, we were able to show for Madlab, Bangladesh. So this proved that the correlation was a very strong one, and again, emphasized the value of satellite imagery for cholera as a sentinel, satellite imagery sensors as a sentinel. This is the work of a postdoc, former postdoc, Constantine de Magny from Montpellier, France, who continued to carry out research as part of the French team in Mozambique, Senegal, and we were able to publish papers showing that the coast of Africa, in those countries, the same correlations could be determined. May I have the next slide, please? So this is a bit more sophisticated model that we're now working with. We have even further sophisticated it, but this was sort of the next stage. We were able to show that warm temperatures 4 to 6 weeks preceding a very heavy monsoon type rainfall in a country with water insecurity, such as Haiti, where an earthquake occurred, or countries like Bangladesh, India, where there are regions of the country with very, very poor sanitation, you have a very high risk of cholera. Have the next slide, please. So we have improved our sophistication of our modeling, along with the ability to use ever increasingly sophisticated satellites. So in 1996, we developed our coastal Vibrio hypothesis, and then we published in 2000 with NASA, with Lobitz and his team in the California NASA NASA station. And then we've essentially moved on to prediction of cholera models with various prototype satellites. And now we're using some of the very sophisticated satellites that measure population migrations and have a resolution of square meters. And so we now can measure pond temperatures, and hopefully we'll be able to measure salinities with some of the satellite, with a satellite that's being launched in the near future. Have the next slide, please. Just to show the application, this is the Hurricane Matthew that traversed from Haiti up the coast of the US. And this was in 1915. We did a retrospective, that is a back analysis of the data of Matthew. And we did a prediction, next slide, of what, based on our model, where the cholera cases would be in Haiti in 2015. That's on the left. And on the right are the actual cholera cases in 2015. We published these data, and we also carried out a study in Yemen in 2017, a retrospective analysis. We have the next slide, please. And we were able to show from the 2017 retrospective analysis, on the left is our prediction of risk, and on the right are the actual cases. This was written up in a squib from our paper, was published as a paragraph in the Scientific American, which was read by a colleague, Dr. Fergus McBean, who is an official with the British DFID Aid Agency, the Department of Foreign Investment and investigation of disease. He rang us up, he called us in January of 2018 and said, could we work together? I like what you're doing. And so what we did then was in fact provide him and DFID, the British Aid Agency, and also UNICEF, our predictions on a monthly basis. And they located their physicians and safe water supplies and medical supplies in the parts of Yemen that we predicted highest risk. May I have the next slide, please? So you will see that in fact, based on our very successful cholera prediction of Hurricane Matthew and cholera in Haiti, and our very successful real-time prediction of cholera in Yemen in 2018, we were able to actually demonstrate a reduction, significant reduction with our colleagues, the British Aid Agency, DFID, UNICEF, the British Meteorological Organization, which provided us with the rainfall and other meteorological data for Yemen, and then our model work at the University of Maryland. And this provided, we now do this on a regular basis. We provide, every 2 weeks, we provide the month ahead prediction for cholera in Yemen, and they use that to monitor and to locate their medical personnel and bring in supplies as needed. May I have the next slide, please? Now, I think I'll go over this because I'll run out of time. We were able to show that the real-time performance of the model using data from Yemen is extremely powerful. May I have the next slide, please? We can show the progression of infection in different climates. May I have the next slide, please? Now, I want to launch quickly into the coronavirus data because what we have done is to take the model that we have developed for cholera and to apply it to COVID-19. The previous slides showed that this translation worked very well. And this is, I think, more assimilable graphically, showing that the risk map is on the left and the actual cholera cases is on the right. This is the early April, early analysis of the COVID that we did applying our model from cholera to COVID. May I have the next slide, please? Let me jump now very, very briefly to DNA sequencing because it ties in very nicely. May I have the next slide, please? I developed back in 2008, 2007, a technique for using whole genome sequencing, next slide, where we extract the DNA from water samples, clinical samples, food, soil, whatever. Next slide, and we run it through the sequencer. It's any sequencer works, Illumina, Ion Torrent, whatever. The raw sequence reads, next slide, are matched up with our exquisite database, about 160,000 curated genomes of bacteria, viruses, fungus, protists, parasites. May I have the next slide, please? This allows us to identify the microorganisms to species and strain. Next slide. And also to identify, because of our gene database, the presence of antibiotic resistance genes, metabolism functioning genes, disease genes associated with pathogenicity, et cetera. Next slide. And that allows us to identify the microorganisms, bacteria, viruses, protists, fungus present. Next slide, please. Right down to strain. And this is very important because as any microbiologist in the audience knows that One strain of Lactobacillus casei is used to make cheese. Another separate strain is used to make Chardonnay wine. Third strain is used to make yogurt. And for the physicians in the audience, a strain of E. coli in our gut very likely provides vitamins, protects us against pathogens, but the strain H0157 is highly pathogenic and causes disease. So strain detection and determination is critical. May I have the next slide, please? Let me give you the example of water safety of this technique, and we'll get back, I'll get you back to COVID-19. So I do have a logical string in my presentation. May I have the next slide, please? We have been doing work with the Los Angeles Orange County Water District, because as you know of the, well, now the fires, but prior to that, the drought in California led to severe water shortages. And the Orange County began to pipe in the water from the sewage treatment plant to recycle for drinking water. And so we worked with them to show each step of the way how purification and removal of pathogens occurred with our technique. So the Q1 water sample is the water that comes in from the sewage plant. The sludge goes out to sea, and the water coming in from the sewage plant is first screened, filtered, treated with sodium hypochlorite, precipitated, microfiltered. It runs through reverse osmosis, filtration, UV irradiation, then a whole series of other treatments, including chlorination. And then the final treatment is to sprinkle it onto a wetlands and then wait 6 months to draw down the groundwater from the wetlands area. That last step, time consuming and expensive. We were able to show that the water that they were sprinkling onto the grasslands is free of pathogens. Next slide. Let me demonstrate that. By our technique, when we looked at the samples of the microfiltration biofilm, you can see the gray area in all the names of all kinds of microorganisms, including viruses, some fungus or two, and occasionally a cryptosporidium. But once it has gone through the microfiltration and reverse osmosis, the only microorganisms, bacteria, that we pick up are the typical water bacteria that you would find in any well-treated tap water. These are iron bacteria, a few perhaps methane metabolizing bacteria, and so forth, but non-pathogenic. Next slide, please. Similarly, we're able to show that as the Q1 water comes in, it has an occasional human virus, adenoviruses, other viruses. It has plant viruses, mainly the pepper mild mottle virus. And then once it goes through the various biofilm treatments, we see a reduction in the viruses to the point where by the time it hits the reverse and gets through the reverse osmosis, very, very few viruses are present. And those that are present are predominantly bacteriophages with an occasional pepper mild motile virus. And we do know that even in tap water, you can pick up occasionally the pepper virus. It's important to know, of course, for agriculture, if you happen to be a farmer growing peppers. Now the next slide, please. One of the concerns was that the sewage water would transmit antibiotic resistance genes. Because we can detect the antibiotic resistance genes, we found that in the Q1 water, the sewage water coming in, lots of antibiotic resistance genes. By the time it goes through microfiltration and reverse osmosis, almost no antibiotic resistance genes, and in fact, not associated with the other than what you would normally find in any soil community of microorganisms. Bacteria would have antibiotic resistance genes that they carry. Next slide, please. Now back to COVID-19. This is a dastardly virus, it really is. as we now know, and in a very short 6 or 7 months, we have learned so much about the virus. It affects the lungs, the liver, the kidneys, the intestines, even the brain. One of the characteristics is losing the sense of smell. It affects the eye as well. Next slide, please. We have been able to determine with our molecular technique, we are able to determine the presence of SARS and its variants. And we can do this, I want to emphasize that the technique we have developed allows analysis from the sample collection. Once we have the sample to the final analysis, we can do this within certainly 24 hours, even getting down to less. The bulk of the time is required for the extraction, library prep, sequencing. Once we have the sequence, less than a half hour, we can do the entire bioinformatic analysis. So this is an extremely powerful tool. May I have the next slide, please? It has been shown by others, this is the publication by Cheung et al. From a gastroenterology journal. The viral RNA can be detected in stool samples in almost every case tested. The various studies shown have demonstrated, there's a whole list of studies, have shown that the prevalence of the RNA, viral RNA, and I'm quite certain these are actual viruses, more about that in a minute, can be detected in the stool. Next slide, please. Now this looks like a very complicated slide, but it really isn't. Vertically, you can see patients, 41 patients. Horizontally, the red shows the study, the clinical sample is positive, and the yellow-orange shows the positive fecal shedding. You can see that long after the clinical test, the swab test, is negative, the individual will continue to shed the virus. Similarly, those individuals who are not positive, we now know can shed the virus in the stool. We have the next slide, please. So what we have done now is to work with Maryland, the state of Maryland, the governor, in a contract with the Department of Environment and working with the Department of Health. These are the stations, the locations where samples are collected bi-weekly, twice a week, at these locations and rapidly transferred to our laboratory. Next slide, please. And we're able to provide a weekly analyses of the presence of the virus in the sewage sludge in the various areas of the state. We're also working with several campuses of colleges and universities in the state of Maryland, being able to monitor specific dormitories and also some nursing homes, so that by sampling the sewage directly from the outlet to the main sewage line from a dormitory, for example, we can detect the virus, and then the college can test the individuals in the dormitory and quickly isolate, quarantine, instead of having to test the whole campus and lose time in detecting and tracing those who are carrying the virus and not yet symptomatic. And also this is valuable because it allows, in this case, Frederick, Maryland, where we're working closely with the health department, it gives them an opportunity to prepare for increased cases. For example, where that peak occurs, one can anticipate 7 to 14 days ahead, cases will begin to appear. So it's a warning for preemptive preparation. It also serves, when the curve drops, that gives additional data for the managers to be able to open up the community. May I have the next slide, please? Now, just as a last bit, let me just show you some data about drinking water. We've been doing metagenomics of drinking water and recently published some papers. May I have the next slide, please? We analyzed bottled sparkling mineral water, artesian water, tap water, and water from drinking fountain in a dormitory. May I have the next slide, please? We were able to show that this is principal component analysis, that the blue is the sparkling natural mineral water, the green are the non-mineral water, bottled water, and the red star in the triangle are municipal tap water, drinking water fountain. So we can determine by the microbiome the source of the water. Next slide, please. Very quickly, the predominant species were really non-pathogens. The typical bacteria that we showed in the treated water from the Orange County, California, analyzed water, the kinds of organisms, and occasionally very, very low numbers of a salmonella, but not sufficiently numerically high to be a pathogen. And we picked up DNA of the opportunistic plant and animal pathogens, and really no fecal indicator bacteria. Frequently, we pick up bacteriophages, which serve as a confirmation because they're phages for the bacteria we detect in the end water, treated water. Archaea DNA was detected, interestingly, only in sparkling natural mineral water. May I have the next slide, please? Finally, let me close. I have described for you elegant techniques of satellite imagery for tracking, DNA analysis for determining, confirming the presence of the organism, lots of other studies I have no time to go into. What good can we do for the villagers in Bangladesh? Next slide. Because the bacterium has a host of a copepod, which is about 200 micrometers, the bacteria are between 2 and 3 micrometers. By removing the plankton and particulates to which the bacteria attach, we can remove 99% of Vibrio cholerae. We've shown that by laboratory work. We showed that simple used sari filter cloth, folded 4 or 5 times, sufficed to provide a 20 micron mesh filter, which would be very effective to remove particulates, plankton, zooplankton, etc. And by training women to be extension agents and go out to the villagers, as this woman, this extension agent is doing, to train the other women how to provide clean water for their families using the sari cloth filtration. May I have the next slide, please? We showed them that it's important to take a yard of sari cloth, fold it, cover the calash, pour water through the calash that filters out the particulates and all the other plankton, etc., but also to rinse the sari cloth before using it again, and especially to rinse it and to dry it in sunlight, which acts as a disinfectant. And this process, next slide, allowed us to reduce certainly the turbidity of the water, made it very clear to the women that the water on the right with no particulate matter is better for their children. And the next slide, we were able to demonstrate 50% reduction of cholera in Bangladesh. We did a follow-up study 5 years later, but it was rather difficult to obtain results because the control villagers had learned about filtration and its effectiveness, and they too were filtering. And so 75% of the villagers in both the control and the test villages were continuing to filter. And we were able to demonstrate, however, a herd effect. Families surrounded by, who did not filter, but surrounded by families who did filter, were protected. May I have the next slide, please? All this work requires collaboration with wonderful people at the International Center for Diarrheal Diseases Research in Bangladesh, the National Center for Cholera and Enteric Diseases in Calcutta, postdocs, students, visiting scientists, especially graduate students at the University of Maryland, and undergraduate students as well. Next slide. And colleagues from around the world, our NASA Ames colleagues from other countries, from other parts of the US. Next slide, please. And water, safe water, as you all are working on, is really a global challenge and remains to be so. Next slide. Thank you. I'll take questions.
Okay, thank you, Rita. That was an excellent presentation. And we definitely have time for some questions. Let's see, I'm going to read them from the chat window for you. Let's see. Oh, Catherine, very interesting.
Everyone is thanking you very much for the talk. Ying Li from Lund writes, great talk. I love your passion and enthusiastic towards several important issues in water. Thank you for saving lives.
Thank you for your compliment.
Okay.
Thank you very much, Rita. That was amazing. Very inspiring. I do have a question, and maybe it's because in the afternoon I didn't quite follow, but when you, somewhere midway through your talk, you talked about how you used your satellite cholera model and used that for COVID-19, and I didn't quite catch how that was done.
And that's a very good question, because I should have mentioned it. I flipped over the slides because I'm worried about time. We have determined that the dew point temperature and the air temperature are significant in the outbreaks. And there are other environmental parameters, including population. And I mentioned that for cholera, we do use, for Yemen particularly, population migrations associated really with, sadly, in Yemen with the civil war. And when you see a mass movement of people around another water area, we can measure the turbidity of the water, the chlorophyll, and so forth, and we can determine the risk of cholera. Similarly, we can, with these ground truth parameters, especially with the very sophisticated satellite sensors we now have available to us, we can measure these environmental factors, and we can correlate that with the distribution from countries around the world. We can get our model to be sensitized, I guess is the word, into these various factors, and therefore provide the prediction. We've just submitted a paper to PNAS, and it carries the details, and I'd be really happy, with fingers crossed that it's accepted, to be able to share it. And we have, in fact, I was up late last night editing another manuscript on the COVID, on population density as a significant factor that will allow prediction during the coming months when people move indoors. In other words, we can measure, we also use cell phone data. That's now woven into the model. So we can measure very, very sensitively movement of people and aggregations of people. And these models are now very, very sophisticated and allow us then to make these kinds of predictions. I hope that helps.
Oh, yes. Wow, impressive. Thank you.
Okay. I have a question here from Stefan Beckerson. In your opinion, well, first, great talk. Thank you. In your opinion, what are the bottlenecks in the metagenome-based pathogen surveillance? How can we do better, even better, use this more widely in the future?
Let me answer this two ways. I think that we've been using sewage testing for about 50 years for poliovirus. I really consider sewage testing a public health tool. And I really think, for example, in our studies with Orange County, we picked up occasional cryptosporidium in the sewage water coming into the, before it was all treated. This is a means of allowing physicians to be alerted as to what's going around the community well before you get peaks in cases. In fact, we show this in some of our papers with cholera, that by prediction of satellite prediction, we're hitting the beginning. It's sort of like any of you follow hockey? Wayne Gretzky? He's a hockey champion player. He doesn't follow the where the puck is. He follows where the puck is going, and that's what we're doing with sewage testing. We're following where the epidemic will be, or potential epidemic or outbreak will be occurring months, weeks, days ahead of time. And so I think this is going to be a powerful tool in the future. Plus, we're able to be, with the sophisticated bioinformatics, be able to even monitor strains of viruses, Now, what is the bottleneck? The bottleneck right now, and we're working on reducing the time. In fact, we now are able to provide same-day sewage analysis to the state of Maryland. And this is by being able to do perhaps either by mobile laboratory or on-site at the sewage station to do the preliminary extraction and just ship, which is very safe, ship the extracted RNA, DNA to the lab. And if we can, and we're doing some studies now with the Oxford Nanopore on-site hand, well, it's not really handheld, but it's a portable sequencer, that we might be able to get this done right at the water treatment laboratory, right at the water sewage treatment plant, and to be able to provide real-time analysis for the operators and to physicians. So I think it's a matter of technology miniaturization for the mechanical part of the bottleneck. For the interpretation, it's really curious that just as I had to take a bottleneck period of time, 20 years, before it was accepted that the Vibrio cholerae is a naturally occurring aquatic bacterium, I have learned from discussion with my colleague, Don Milton at the University of Maryland, who has been working on airborne transmission, that there is ironically a similar bottleneck for physicians to accept airborne transmission. We're getting to that point because we at first, physicians are taught to wash their hands, to wear masks at the direct site, large particulates, but this micro droplet floating transmission hasn't been really accepted. It's beginning to be accepted. So I think it's, I think there was a philosopher of science who in his book said that it takes 20 years, but I think it was paraphrased to say that no, what it takes is for the naysayers to die for the acceptance of the new paradigm. Anyway, that's an extraordinarily long answer to a short question.
Okay. We're kind of moving in an interesting direction here with that answer. I would like to take a question from Catherine Legrand. She asked, how did you approach politicians and policymakers maybe you can say a few words
Yes.
about this.
First of all, with respect, and I worked with both sides of the aisle, but then at a time when you could. I was a director from 1998 to 2004, and then have done some policy work since. I think my assumption has been that politicians who disagree with whom I may disagree on specific issues, nevertheless, are fundamentally and basically, for the most part, they're patriots just as I am. And their fundamental desire is to improve the situation. They come at it differently. But if we can compromise, then it benefits both of us. And that's pretty much how I've been able to succeed. For example, my prime example is Newt Gingrich, with whom I disagree on quite a few fundamental things, but he loves science. And he and I get on extremely well. He was instrumental, particularly during my years working under President Bush too, in helping keep the NSF budget high. I managed in my 6 years to increase the budget by about 67 or 68%. I didn't quite double it, but I almost did. And that was because of working with both sides of the aisle.
Okay. I have another interesting question here about if you were to start a career in this field and want to make a real difference in this field, what should you study?
Well, my granddaughter, who is 18, is taking as I mentioned, is taking a gap year, wants to study neuroscience. And I applaud her because I think decoding the brain is going to be amazing, but it has to be done with very careful ethical boundaries for artificial intelligence and for, well, machine learning in general. But I think the marriage of computation capabilities with the extraordinary discoveries in biology are fabulously exciting right now.
Okay.
And interdisciplinarily focused. I would like to emphasize that needs to be tied to the social, behavioral, and even the economic sciences. We have left out the social behavioral sciences in our natural science research, and we need to tie them together.
And how do you propose to do that?
I think by one trick that I used is when I was making a massive investment along with the extremely good help of Neil Lane, who was science advisor to Bill Clinton, in providing about $1 billion new for computer science. I made sure that every directorate, including the education directorate and the social behavioral science directorate, got a portion of the funding for two reasons. They needed to incorporate computational sciences, but also to bring them on board and not have them complain because the bulk of the money would be going to the computer science directorate. A little bit of psychology.
Excellent. I wonder if you could take a few minutes to talk about your book that just came out.
Sure. I did not start out to write a book about women in science. I wanted to write about cholera because I consider that story exciting. And the book actually lacks 2 chapters. One chapter, I just didn't have time because Simon & Schuster wanted the book this year to do a chapter in oceanography because I had some fantastic students, Jody Deming amongst them, who is now a member of the National Academy and a professor at the University of Washington, and a number of other extremely good students who we worked together developing deep sea sampling devices, et cetera. And the other is the international collaborations. I've worked beautifully and with great joy and fun with my colleagues, Carla Pruzzo and her team in Italy, with my Monique Pompuy and colleagues in France, former students and colleagues in England, with former students in Japan, with former students in South America, in Australia, I mean, all around the world. And I wish that that chapter had gotten written, so maybe I'll have to do that. Anyway, I got a phone call 5 years ago as I was thinking about the book on cholera from Sharon Birch McGrain, asking if she could write a book about my work on cholera. And I said, sure. She said, let's collaborate. I said, fine. So as we started writing about the steps that I had to take in moving from discovering the bacteria is an aquatic bacterium to all of these other discoveries where I was considered a loony, I found out that famous women and not famous women, but very good scientists who never got recognized properly, went through all of the same trauma. And so it ended up being a book more about women in science and the traumas that they had to go through and the barriers and the good fortune of having a family, a mother or a father who was very encouraging, as my father was. For him, it was a no-brainer for women, for me as a girl and for my sisters to go to college. Sure. And then to have the extreme good fortune of marrying the best guy in the world that I could have, whom we essentially decided to get married on our first date. And we were married for 62 very, very happy years. I miss him every day. So that was another good fortune. And then to have a wonderful PhD advisor, mentor, John Liston, who was a Scotsman newly arrived at the University of Washington, whose motto was that rules were made to be broken. He broke a bunch of rules to get me as PI, as a grad student, or really just finished in a newly minted PhD, co-PI on an NSF grant, and then appointed a research assistant professor at the University of Washington, and then granted a leave of absence to go to the National Research Council of Canada, because both my husband and I had received postdoctoral fellowships from the Canadian government, but I got a second letter saying, oops, women, husbands and wives cannot both, because of nepotism rules, which now has been changed, receive fellowships. So Jack got it, but I didn't. But John Liston's reaction was, well, he said, there's more than one way to skin a cat. And that's when we applied for the NSF grant, et cetera, et cetera. So there are really good guys out there. You just have to find them and team up with them.
And so you've made a tremendous efforts at the NSF in your policy work and increasing budgets. Do you think policy alone can change the system?
No, the system has to change. We women aren't broken, the system's broken. As the Me Too movement shows very strongly, when you speak up and speak out, and when all speak up and speak out, finally, the voice gets heard. And I think it was fabulous that I chaired not fabulous that I chaired, but fabulous that the report from the committee that I chaired came out just at the peak of the Me Too movement. So here were all these voices, and underpinning it was this 1-inch thick, data-driven report, not full of whinging and gringing, but just facts about what happens when an application that's identical, but is Jane or Jennifer, and an identical one is John, how differently they get treated. This was the Joe Handelsman's experiment that was published in PNAS. So it was really gratifying to have this mountain of data from a committee of fantastic women. The co-chair was the former secretary, the first woman to chair to be Secretary of the Air Force at a time of tailhook when there was egregious sexual harassment of women by the Navy pilots. And Dr. Johnson, an African-American female physician, president of Wellesley College as a co-chair. And then Connie Morella, former 20-year member of Congress, just a staggeringly great team on the committee, giving this report at the time women were crying out, saying, look, I have been really, really badly treated. In my view, the book provides the iceberg. My comment is, yes, it's from the criminal to the clueless, a whole lot of clueless under the iceberg, and the criminal is what you see in terms of physical attack and sexual raping and that sort of thing. That's the criminal, but way down is all the clueless behavior that makes life miserable for us.
So if you could say one recommendation moving forward.
Stay the course. If you're interested in STEM career, damn it, do it. There's a way around, under, behind, over. That's barriers. Your path may be zigzag, but it's just as good when it gets there.
Great.
Just look at some of these football players. When they make touchdowns, they don't go in a straight line. They knock out a lot of people on their way to get there. So
great. Thank you, Rita, for a very powerful, positive voice. Appreciate your time. I know you're running to another conference.
Yes.
So I really appreciate that you came and spend this time with us and have these personal reflections.
Well, thank you. My only regret that I'm not there with you in person.
Yes, next time.
That would be fun. And till we meet again, and go forth and do good, all of you.
Okay. Thank you again, Rita.
Bye-bye.
Okay, bye.
Bye.
Okay, so we will continue on. I just want to take a step back and reflect a minute that prior to the current pandemic, and as Rita has reflected here, there have been challenges in women achieving gender equality in academia. And since this pandemic has hit, it's been hitting scientists, men and women very hard, and in particular women. And here in Sweden, there's some work being done to increase equality for women in academia. And so now I'd like to introduce Maria Saline, a biochemist by formal training, and she's the project manager of the recently started GENIE program. So GENIE stands for the Gender Initiative for Excellence, a 10-year program funded to create a more equal gender balance and better working environment at Chalmers University. And Maria is going to provide us a brief overview of the GENIE program, and there'll be opportunities to ask questions. I'd also like to take this opportunity to also introduce our invited speaker for tomorrow, Lauri Robbins Ericson, an international expert and consultant on emotional and gender intelligence. So welcome to both Maria and Lauri, and I'll let Maria take over.
Thank you, Rachel. Wow, I'm almost speechless after listening to Rita. What a power woman. I'm super impressed. Thank you for inviting me. I'm here to present to you the GENIE project, and I'm going to share a few slides. So as Rachel said, my name is Maria, and I am a biochemist, so it's been very interesting to listen to all this about bacteria. But then I took a detour, so now I'm not a biochemist on a daily basis. I'm doing my more project managing work. Now let's see if I can share my screen. I hope some of you have heard of this GENIE project. That's a big part of why we're doing it. But basically, it's a Chalmers. And why? One can ask, why did GENIE start at Chalmers? And I think mainly the reason is that we're the worst. If you look at the numbers, Chalmers has the least number of female professors. It's not good anywhere, and this is a comparison of different faculties, not just STEM. But Chalmers is the worst. Oops, sorry. And I think that's what got the Chalmers Foundation to actually grant the application that was written by the faculty. So Chalmers is a foundation university, so we get money from our foundation. And they had a great call and said, we need to do something to improve the university. Faculty come up with ideas. And the 3 ideas that were granted was GENIE for gender equality. There was one about AI research and one about improving the teaching, both the rooms and how to teach. But the GENIE project is what I'm working with. This is a statue that is outside Chalmers. We're very proud of that the first female engineer was examined at Chalmers in 1917. There's 500 men and her, and this Vera Sandberg, who's her name. There's streets after her, there's rooms after her, and now we also have a nice statue of her doing some chemical labs work. So why? I think Rita started talking a little bit about why is GENIE needed? Why do we need to think about gender equality? And why do we still, 2020, keep working on this? I was listening to Nanna Gillberg, and she said, if we don't do anything, it'll take 186 years until we're fully equal. And we don't have that time. So we just need to get something going. And I think you know most of these why, but we constantly have to repeat. As Rita was saying, there's so many of the men and women that are just clueless. We don't know. And you live in your box, and this is what you see. So we need to make people aware that there's still biases. It's not fair game. Women are not treated the same as men. There are many other minorities. Women are not minorities, but minorities that are not treated as well as the white men either. So all these biases, you just need to constantly say, it's not perfect yet. So there are systematic biases. It's harder for women to speak up because we're so connected to our network and we're afraid of speaking the truth sometimes. Chalmers is the worst. And if you look at the data, women are being more sick. I think that's the same in all of Sweden, but it's a sign of something is not right. We also know from a lot of research that mixed groups, and Rita was talking about that we need to do interdisciplinary focuses. Women, and we need mixes of groups to do better science, to be more innovative, for companies to do better profit. It's a no-brainer as well. We need to have mixed groups to be a beneficial and good university. And to reach that, we need to have role models that are female, and industry wants to have female engineers, and they want to have men that they know about gender equality. So we just need to focus on improving everyone's knowledge so we can hire the best people and keep them. So what is GENIE? It's, we say, a strategy to increase the success at Chalmers. We have quite a lot of money. As far as we know, it's the largest initiative in academia. We have 10 years, so this is our second year. And I got tossed into this project in January 2019. I was new at Chalmers. I just left my postdoc at AstraZeneca, and I thought, well, I want to learn about more behavioral stuff, how people work. I'm tired of being in the lab. I want to work with people for a while. So I got tossed into this project, and it's no lab at all, but there's lots of good people knowledge. And it's led by faculty. So we have 10 years, we have a lot of money, and it's led by faculty. Those are key points, because most other initiatives are led by HR or the management. And we know in academic setting, the faculty, it's a bit different, but this is the faculty doing their own work with help of HR and project managers like me. But it's two different languages to speak, so to say. Our goals are to have a gender equal culture and systems. This is really tricky. The culture is us, all of us. The systems we keep working on, not easy either. And we want to increase the female faculty to about 40%. And that means also all the levels below should be at least 40%. And there should be no salary gap, for sure. So how do we do this? We have 3 main areas of work. One is measure and analyze, because I mean, we're scientists, we need to know the numbers. And it's also a way to get people not talking about their individual history and what they've been through and sort of n equals 1. Everyone has some experience, but we need to have the data so we know what we're talking about. So we measure lots of data gender divided, and I'm going to show you a little bit of that later and show you can find it on our website. We did some bibliometric studies. That's to look at all the publications. That's trickier. We didn't do too much of it because it was complicated, but it's something we are going to look into more, how to have that gender divided. We have a fellow PhD student who follows most of what we do and is going to write a couple of social sciences articles on the GENIE project in comparison to other initiatives. And it feels a bit strange to have someone looking at us, but we see this as a big experiment. So we need to do experiments on ourselves and kind of monitor along the way. We do employee surveys, or we of course have some KPIs, some key measures for the foundation who gives the money to us. They need to make sure that we're doing some progress, but it's very hard to measure culture, and it's hard to measure how people are behaving and how you kind of perceive things. So what we do is some central actions where we have created a couple of supports of how to support the faculty that's here, but also how to recruit new faculty, visiting faculty. We do education information, and we try to work with awareness and the policies. But most of the effort is actually put locally at the different departments. Chalmers has 13 different departments. So we try to somehow make tailored recommendations for each one of these departments. There are 13 different cultures and maybe even subcultures within these departments. So there's lots for us in the leadership group to understand and learn. I haven't worked at Chalmers before, so I didn't know how it looked. And the leaders are from different departments and they mainly know their departments, but we have to see all 13. So we have one person for each department that is the GENIE representative, and we try to work with them more in detail. So it's kind of a combination of female recruitment and support, and also the long-term, more systematic work for a slow cultural change. So it's both top-down and bottom-up, and we need to get the majority on board. It's not a fast work, but it's really exciting. The leaders of GENIE is Pernilla with the dark hair, Vittung Stafsede. She's a professor in the bio department. And then the vice leader is Mary Sharon. She's a computer scientist. And down in the left corner, you can see them giving speeches. Pernilla talked for the KVI and Mary at some coding conference. They're both lovely people and very passionate. And we also have in our leadership group 2 prefects from different departments to get their sort of the management's view on how to implement things. We want to make it the structural changes. So we need to have them on board. And then we have some other stuff that is needed, economy and HR and stuff. We have an advisory group. And here we particularly have taken a lot of advice from 2 of them. Paul Walton, who is a professor in chemistry at the York University in UK. He's worked with gender equality in academia for more than 20 years. So he's a very good and experienced person to talk to. So we have actually in these Corona times, we had weekly meetings with him. It's been lovely. He suggests we should have GNTs together, but I think that will be in the future. And Lisa Husu, she's a gender professor from Umeå University who knows all the studies and can give us advice on how to use the data, what data to monitor, and also just what is known in the field. And then we have Agnes Wold from Gothenburg University, I think you all know of her, and 2 representatives from the industry. The steering group is Stefan Bengtsson, who is the rector, the president of Chalmers. So we have connection to the top, which is very good. And he's passionate about this. Chalmers is going through some economic issues. We need to cut down on people. So there's both COVID and economic crisis. We're not in a good situation. So Stefan is working hard, but we're constantly reminding him that this is a big worry for the gender equality. Most of these times when you have to cut people, you have to make more work. Who's going to do that more work? It's usually the women that raise their hand and do it and get nothing for it. So we're on him to make sure he does not forget the gender equality in everything he does now. And then we have 4 more people, the head of the student union, the HR person, because there's a lot of HR issues. So HR head is part of the group. And then 2 wonderful old professors. So what have we done? This is a picture from the Chalmers campus. Quite fitting. This machine is called a GENIE. So central actions. We have, the first thing we did, what we bought, or we didn't buy, but Chalmers has a big open selection for research assistants. And in that call, I think there were 1,200 people applying for 10 positions. And after all those 10 were taken, we took number 2, if that was a woman. So she was highly ranked and a very, very competent person. So we hired 5 assistant professors. And then we also made more of a general package. If some department want to hire female assistant professor, this is what GENIE can provide as a starting package. But it has to be a highly ranked person, and in there should be a big open call for that. We also made guidelines to support direct recruitment. So Chalmers may directly recruit people. So this is a benefit, but it's not easy to find people who want to move their group or maybe want to move to Chalmers. But we have 2 ongoing cases, and these are 2 women that actually heard of GENIE. Through Pernilla and Mary are often talking about GENIE when they do their ordinary out speaking at conferences. So we have 2 ongoing discussions now of very nice, good female professors. So hopefully they will be directly recruited, and then GENIE will be part of their started package. So we work a lot with the head of departments to make sure we can provide for the people to come. We have a female visiting faculty program. Since Chalmers is in economic problems, we are not allowed to hire people except for some specific direct recruitments that may go through anyways. But it's hard to hire people. So a visiting female professor, it's easier, and we will still have someone on campus that is seeing what Chalmers is like. She can be a role model, she can inspire some students, and we can get more network going. And maybe in the future that will be a recruitment. So far we have 7 approved and 1 is already teaching. So that's nice. We can pay if you want to go to a gender conference, and we had an internal funding. So it was an open call where we said, we will fund projects up to £1 million if it's coupled excellence to genders. It's a quite broad and undecided field. So in the reading all these applications, we took help of Lisa Huso and Paul Walton, and then 7 different professors within Chalmers. And we ended up getting 30 projects that we funded. We had 78 applications, 30 projects which we're sharing now 23 million crowns. And it covers all the departments as Chalmers. So this is really bottom up, very different ideas, and exciting to see what they're gonna come up with. They have another year to go. And then we'll do some big sort of combining all their data and see what happens. So measure and analyze. I told you we have a PhD student that follows us. We're doing some bibliometry. And we also gather a lot of data. what is it like at Chalmers? Who, at positions, sick leave, salaries, those data that are in the databases, we are showing them openly on the website, and also for the head of departments to know. Of course they know it, but it's better to see other departments as well so you can compare. And also for the employees to see what is it at my department compared to another department. We will address academic household work or good collegiality, whatever you want to call it. It's more complicated. We're looking into the money flow. This is also a complex system, so it's good we have a finance officer in our group. But you probably all know, you get your salary from certain project, but maybe you do something else, or maybe you do several things in that project. So for us to follow the money and actually get data that you can trust is not easy. We want to see the workload, who is doing what work, but getting that data it's not super easy. And to understand the culture and to follow how culture is changing, we have designed a few special questions for the yearly employee survey that Chalmers is doing. So this is something we'll go back to every year to hopefully find that it's being improvements. So I just want to show you two of the data from this gender data that we have for 2018 and 2019, and you can find it on our websites. So here you see how many female are at each position at Chalmers. So it's quite low as in the full professor. Chalmers has a professorship that is biträdande professor, so it's almost professor. There's a few more female there, and we're going to look into why the women kind of stop the step before becoming a full professor. And these assistant professors, here's the only category we're doing fine, and mainly a lot of it is because we added these 5 extra in the open call last year. What is very worrying is the sick leave. Female are the orange and the male are the green. In general, people are not so sick, and you know, we're very free at work, and many people work from home even if they're sick. But even so, this is all over, female are more sick than male, except for in the TME department, which is very progressive. It's management and economy. They have had a very good prefect for many years that worked hard on gender equality. So here, the women are happier and the men are starting to realize, whew, I gotta work hard. I can't have smooth sailing being a man. That might be what's showing here. But it's interesting to look at this, and it's nice to have the data so we know we can follow it from year to year, and you can also kind of have a common ground when you're talking. But the most important thing we do is working with the departments. This is where the action of changing the culture and getting it to stick will happen. So to help them, we have a GENIE representative. We've created a toolbox, which is a document where we just put up everything we could think of what to do. It's anything from a small looking at statistics to actually fire the people you want to get rid of. So it's a spectrum. We also requested that every department will make a gender equality plan and include it in their operational plan. So they did that last year, and we'll follow up this year. And to make a little bit more incentive, we promised 2 million crowns for 5 years for this gender equality plan. And there's of course constant gender equality work being made, the jämställdhetsintegrering, the gender mainstreaming, but this should be a little bit on top, a little bit extra to give it a push. So this is where we spend most of our effort. But we also do a couple of small things, bottom up. If people approach us with ideas, for example, the WISE VACT, there are 2 networks for female students in the electronics and nanoscience. They wanted to do a mentorship program and needed some help with administration. So we're paying for an administrator. The students started a women association. So we are giving them some funds so they can get going. A male professor approached us and said, why are the female leaving from chemistry? It doesn't make sense. I want to investigate this. So together with Lisa Husu and a scholar, he's looking into what is it like at chemistry. So doing basically exit interviews and surveys, going back and see people who left, why. So this will be a lot of good data for us to know. And it's really exciting when there's a man who comes to us and says, this is something wrong. What can I do? We do events, seminars, both internal and external. And as I said, Mary and Pernilla are often out speaking at conferences. We try to have some informal gatherings, some fika, some lunches. And in these Corona times, it's a lot of meetings on the screen, but I feel like there's been more of them, and a lot of people want to kind of linger on after the meeting and just talk. So this is good, and we get ideas. We've spoken a lot about what to do in these COVID times. Women are suffering more than men. How can we use the GENIE money and our power to do something? We have built a gender equality literature based on Lisa Husu helped us to do this. And we have more stuff on our website. It's constantly being grown. And then a lot on my side as a project manager is that I talk to the other people at Chalmers who work with gender equality issues. Fokus Jämställdhet is a forum for everyone who has something to do with gender equality at Chalmers. It's good to meet them. It's the pro-rector and the vice rector who is in charge of this. And we talk a lot with the hiring committee. And we are training ourselves, and we'll make a course on cultural and how to change work environments to educate these GENIE representatives, because they are key people. They're sometimes the head of the department, and sometimes they're another quite high up person. So there are many things going on. And I think these 2 years we've been working, we have built a lot of trust. There's a lot of unhappy people in the beginning, and 300 million just down the drain. Is this the Chalmers I want to work at? But we worked lots and showing that we work with them. It's not us telling them what to do. We're not the management. We are part of the faculty too. We're listening to them. We want to do it the academic agenda and then change slowly. And it's always the excellence. It's always the meritocracy. That's going to be who we choose to hire. And it takes change, and any little small action is important. Paul Walton keeps telling us to be generous and to listen to anyone who has a small idea. So that's why we give money to many small little things, and it's exciting to follow them up. So that's where we are, and that's what we're doing. And it's really, really fun. I'm going to stop sharing here, and I'm happy to take some questions.
Excellent. Thank you so much, Maria, for giving us that overview of this very important new program at Chalmers. Excellent. I read a few questions from the audience. So I have two from Eva Sorensen. She says, interesting. How do you include students, for example, PhD students? And then she had a follow-up question with, do you interview current employees, both men and women, on how they experience their work situation?
Yeah, we don't interview. We want to do that, but it's just such a big job to do that. We're trying to make the departments do their work, but that's something that maybe these GENIE representatives, we're giving them 2 million. You can actually hire someone to do the job for you. And many of them are talking about doing some cultural surveys, which would include interviews. So I think it's going to happen. Things are just very slow. And also with this COVID and then economy imbalance, it's very hard to make something happen. So we are trying to be very patient, but I'm thinking that will happen for sure. And regarding students, we do meet some students, and we have given this Chalmers Women Association our students. We pay for a few when they want to have someone come and give a seminar. We said, yes, that's great. We can pay for that. And if you do that, you need to open up so everyone can come and listen. But mainly the faculties are an audience. PhD students, we're including more and more because we see they are suffering a lot. And I remember from my PhD, so you're very vulnerable as a PhD student. They're not our primary audience, but definitely we are talking to them. And so we have a constant dialogue with the student union.
Okay. So I'm going to try to be mindful to our program that is ending according to our schedule now. But I really want to encourage everyone to come tomorrow. Maria will be on our panel tomorrow. We'll start the day with a presentation by Lauri Ericson, and then we'll move into an open panel with several of the speakers today. So I want to encourage everyone to please return tomorrow for part 2 of this conversation about gender equality and emotional intelligence. And as we discussed between Anna and I, to follow up from her survey this morning, or it wasn't even this morning, this afternoon, she's going to show again her Menti survey, and we are going to ask the question about what clean water means to us. Correct, Anna? What connects for you safe water and microbial ecology? So we'll see how our answers have changed. I want to thank our coordinator again, Carolyn Littlefield, for her help, and also Anna for sharing this day with me as my co-chair. It's been an excellent day. And please do come back tomorrow at 9. Those of you in our AMRI network, we look forward to continuing these conversations. I just want to take again opportunity to thank everyone. And Lauri, if you have anything you would like to say to prepare us for tomorrow, you're welcome to unmute yourself.
I do have one quick thing.
Okay.
And being mindful of everyone's time. For those of you who are participating tomorrow, if you could, because I work on the behavioral side, if you could think of one person in your life who you consider to be a leader. So someone inspired you to do the work that you do or be the person you are.
Okay, excellent.
And we will talk more about this tomorrow.
Okay. So I think we'll call this meeting to a close. I thank again everyone for being here. Thank you to the audience for spending the afternoon with us. And thank you again, Carolyn, for making all of it work. Bye.
Bye. Thank you.
Bye. Thank you.
Bye.